pharma faculty members under the SERB-CRG grant, Social Scientific Responsibility Initiative. We have faculty members from different states of the country, and we had to limit the participation number to 25 from 62 applications, which we got within 3 days of posting the announcement on different platforms. I'm glad to see you all, and I have got high hopes for what we can achieve while we are going to be joining here. Coming away from our day-to-day work is important every once in a while, and I know that it will be difficult to relax about what's going on back at the office. Please keep your mobile phones on silent mode, and if you have an extra laptop, that also I request you to switch it off. There will be set times during the day we will be able to check in at work. The main objective is to focus on recent developments in cancer, which can help young researchers and middle-aged researchers in the field, and also to enhance the skill of writing successful grant applications and have a promising research career. There are certain expectations from this workshop, and we all will be benefited only when we participate with open mind and share our ideas and thoughts with the eminent group of speakers and participants. We are here so that we can improve our skills. Not only so, but also to identify key areas from the sessions to move forward and resolve challenges in cancer therapeutics. The schedule has been shared with you all for both the days, and I am quite hopeful for the timely conduction of this workshop. On this great occasion, and on behalf of SPPSPTM, SVKM, and NMIMS, I welcome all the faculty participants and request our respected Dean, Dr. Bala Prabhakar, to address the faculty gathering.
Thank you, Dr. Moshmi. First of all, a very good morning to all the participants and our speaker, eminent speaker, Dr. Shilpi Dutt. I would like to thank her for accepting our invitation forwarded by our faculty members very quickly in such a short notice. Thank you so much.
I'm honored to be here.
We are very happy to have you here. And first of all, SERB it's a new initiative nowadays. This is coming as a social responsibility. The government is mentioning that whatever research that is happening with the scientists and the grants that they are giving. In this case, our faculty, Dr. Mayur Erigiri and Dr. Moshmi Kumar, these are the recipients of the grant. And as part of the social responsibility initiative, they are supposed to be conducting workshops to benefit the other faculty members also in the area that they are researching, which will also trigger the thinking and trigger the collaboration. That is the main purpose that the government is doing. And aptly to that activity, to that end, our own faculty members have started this initiative. As Dr. Moshmi has mentioned, quickly we could get the participants. And in fact, I was asking Dr. Mayur to open it up for the entire country, but then they all felt that it should be individual interaction and should be benefiting each person, and there should be interaction between the faculty members so that it leads to some impact and innovation and improvement in their research areas and also collaborations. So that is why we had restricted the numbers to 25. I'm sure you will be all interacting with each other and benefiting from this. And since this grant that is received by our faculty members is in the area of cancer and resistance, in which Dr. Mayur has been working, I think, for nearly 2 decades. Isn't that so, Dr. Mayur? So they have also chosen the topic aptly, cancer research. And as you know that worldwide the cancer is going on increasing. Though there is so much of research, still we cannot say that, yeah, if you have cancer, it can be cured. No, it's not so. So all of us, all the faculty members will have to contribute more. And it is such a tough disease and such a widespread disease. Even though you can see Dr. Mayur, still he is trying to take molecules to the market, so much of work. It needs a lot of time. The point is it needs a lot of time, lot of effort. So the first topic that they have chosen for day 1 is cancer research, which is very apt. And we have all eminent speakers, not only from India, from organizations like ACTREC, eminent speakers, and also from IIT Gandhinagar, and from Bangalore, as well as from Netherlands, where Dr. Mayur has lots of collaborations. And let me tell you, all the other speakers also are most of the time collaborators for our own faculty members who are carrying out research in these areas. And for tomorrow, again, we have innovations and applications where most of the time we see that our faculty members, when they are submitting the grants to different agencies, the grant applications get rejected. So the teachers get disheartened and they feel I'm continuously applying, but it is getting rejected. So this is one opportunity they are actually taking to discuss one-on-one also and understand what is it that the evaluators are looking for, what is it that should be there, and it will be an interactive session with our faculty members, with faculty members that are participants, and also the faculty members who are actually receiving grants at national as well as international level. So it's going to be a wonderful session for these 2 days. I hope you will be interacting and you will be there in the entire session. It is total scientific sessions for today, and I hope you will enjoy and get good positive outcomes. And I expect at the end of the session, there will be a lot more collaborations between the faculty members who are participating as well as with our faculty members also. So I once again congratulate our team at SPPSPTM for taking up this initiative and quickly putting together this activity, and also best wishes for this entire program. So enjoy the science, enjoy the learning, have a good day, and also stay safe. The cases in Mumbai are increasing, COVID cases. So stay safe, please take care of your health. Thank you.
Thanks.
Ma'am, you are on.
You are muted. You have to unmute.
This is going to happen on online platform.
Yes, I know. I'm worried that my slides will be shared.
Yeah. Thank you so much, Bala ma'am, for such encouraging words at the beginning and opening speech of yours for today workshop, talking about the genesis, academic and research background for organizing this workshop. And you really put it very well. Thanks a lot. And before we move towards the scientific proceedings for the day, I would request all the members to please keep your video camera on so that we can quickly click a picture for our records. That would really help. Yeah. Good to see new faces from all over the country. Dr. Jaya, Dr. Sangeeta, and Dr. Khushwant. Yeah.
Dr. Khushwant is still traveling.
Yes.
Okay. Kapil is not there.
Yes, ma'am, I'm taking picture. Just a minute.
Okay.
I'm there, ma'am.
Okay. Let me know once done.
Yeah, ma'am, it's done.
Thank you so much.
Thank you. Thank you.
Now we will go ahead with the scientific sessions for the day, where we are going to focus on biological aspects, newer targets, drug resistance, cancer biomarkers, models to study drug resistance in cancer. As we all are aware, cancer therapy faces a major problem due to failure associated with acquired and intrinsic resistance mechanisms. Around 7.76 million cancer deaths occur every year worldwide. The most characterized mechanisms of resistance are altered cellular drug transport, increased survival or decreased cell death, altered DNA repair, and alterations in drug targets. Today, we have privilege to have a best scientist to talk on the subject of biological causes of cancer and epigenetic regulation of DNA repair in cancer therapy mechanism. Of resistance. Dr. Shilpi Dutt is an esteemed scientist at Tata Memorial Center at Actric, Mumbai. Her research area is therapy resistance in cancer, DNA damage repair phenomenon, epigenetics, glioblastoma, and leukemia-related cancer. She has immense experience in the field of cancer research, and her focus is on gaining insights into the radiation and chemotherapy-related resistance, molecular mechanism in cancer, epigenetic regulation, and the DNA damage response in malignant conditions. She has published numerous articles in internationally reputed journals, such as Cancer Letters, Journal of Cell Science, OncoTarget, and many more. If the participants have any question during the session, you all are requested to put it during the session itself in the chat box so that the speaker can address it during the lecture or towards the end of the session. We are going to follow the same protocol throughout the day and even for tomorrow. And the lecture time for ma'am would be 45 minutes, followed by 15 minutes of interaction with the faculty participants. Let's hear from Dr. Shilpi Dutt.
Thank you, Asmi, for the nice introduction. And I'll start by thanking the organizers for this opportunity where I can present my work. And I hope to get new collaborators over here. Let's see if I can share my slides. I'm just going to share my screen. You can see my screen?
Yes, ma'am, it's visible.
Okay, I'll just put it on the slideshow. Why is it not going to the slideshow? So it's okay, right? It's on the slideshow?
Yeah, perfect.
Okay, and like I said, I would want to thank the organizers for this opportunity. I'm Shilpi from ACTREC, as Maushmi has already introduced me, so I'll not take further to introduce myself. So today I'm going to talk about mostly about the work that we do in the lab related to epigenetic regulation of DNA repair in cancer therapy. And I'll give a very brief introduction about cancer because as I understand, most of you would be very familiar with cancer and working on different aspects of it. So cancer, as we all know, is uncontrolled proliferation of cells where the cells actually have the ability to invade and metastasize. And it's resulted because of the alteration in the gene structure or the sequence, gene expression, or epigenomic alterations in the genome, which means that cancer is basically a genetic or a genomic disease. Now, because of the vast information that we had about cancer in the field, so in 2000, Hanahan and Weinberg came up with 6 hallmarks of cancer. And this was because the information was very vast and very diverse. So it was a confusion in the field as to when do you call a neoplastic disease as cancer. So they came up with 6 hallmarks of cancer, which together constitute an organizing principle that would provide a logical framework for understanding this very diverse kind of a disease. And majority of these, sorry, I'll just have the point. So these hallmarks include that the cells should be able to sustain the proliferative signals. They should be able to evade growth suppression. They should activate invasion and metastatic network in the cell, enabling replicative immortality, inducing angiogenesis, and they should be able to resist the cell death. However, after 2000, there was again a huge surge in the technology that we have now in the field, and because of all the genomic platforms that we have, a lot of new information was added to these, which led Hanahan and Weinberg to come up with another 5 hallmarks of cancer. So in 2011, they came up with this paper where they say where it is the hallmarks of cancer, the next generation, and they added 10 hallmarks of cancer, like 4 new hallmarks of cancer, which included avoiding immune destruction, then tumor promoting inflammation, genomic instability and mutation, and deregulating cellular genetics. Now, energetics, that is the metabolism. So now, I feel that genome instability and mutation should have been there already before, but you would appreciate that most of these genomic, like after 2000, there was a lot of newer techniques that actually gave us a lot of information about the genome and the mutation, which was not there before. So that's why this genome instability and mutation was not included before when they came up with the 6 hallmarks of cancer. So genome instability is the fundamental of any cancer, like DNA damage and DNA repair are the fundamental pathways that are involved at every step of carcinogenesis. At the genesis, during the process of the progression of the cancer, and also, as we'll talk about, in the therapy resistance as well. So DNA damage from its earlier stage, cancer development is associated with DNA replication stress, which leads to double-strand breaks, genomic instability, and it leads to the selective pressure for p53 mutations. As you can see in this schema here, when the damage is induced in a cell due to exogenous or endogenous sources, in a cell that have a proliferative capacity, ideally either the DNA gets repaired, or if because of the errors in replication, the repair doesn't happen, the cells should undergo cell death. However, in the case of cancer, if there is a problem with the cell death machinery or with the DNA repair, these cells, they accumulate mutation, which confers a selective advantage for the clonal expansion, leading to pre-malignant disease, and then finally to the cancer. Major modalities for cancers include surgery, chemotherapy, radiotherapy, and targeted therapy. But of all these, after the surgery is done, chemotherapy and radiotherapy are still the major modalities, and targeted therapy is targeted and it is limited to based on what kind of genetic background the patient has. And because it's expensive as well, so not all the patients can afford to have a targeted therapy. However, chemotherapy and radiotherapy still remains the major therapy for the cancer treatment. Irrespective of the genetic background of the cancer, it can be given to all the patients, and they are not expensive as well. And the major goal of chemotherapy and radiotherapy is to induce enough damage to the dividing cells, that is in this case cancer cells, so that the DNA damage response of the cell is tipped towards apoptosis and the cancer is stopped. So because it is involved, it basically mediates damage to and kills the cells that are highly proliferative. That's why you see that there are side effects of hair fall because of the stem cells that are proliferating in the hair follicles, and also some problems with the intestinal, because those cells are also very replicative. However, that said, the major problem of resistance still exists. So cancer cells are really smart. And some of the cells, not all of the cells, but some of the cancer cells, they, because of the genetic variation or manipulation of this DNA damage pathway by genetic regulation or other modulations, can resist this damage, or they can survive the damage, leading to the therapy resistance, which is still a major clinical problem. If there was no therapy resistance, then I think radiation and chemotherapy are good enough to take care of the tumor. So I have highlighted this portion in red because this is the part where our lab focuses on, and this is the interest of our lab. So to address these questions, we have two major models that we use for radiation therapy and for chemotherapy: glioblastoma and leukemia. For today's talk, I will focus on glioblastoma. So glioblastoma is the most fatal primary brain tumors. As you see here, this is how the CT scan of primary GBM looks like. This is the tumor over here. And tumor is surgically resected, followed by radiotherapy. And following this radiotherapy and resection, the patient is sent back because the tumor is gone and tumor is not identified. This is post-treatment. However, as we know, that after a few months, there is a recurrence of GBM. And this recurrent GBM is very aggressive, highly aggressive, which means that because of this aggressive nature of GBM, the median survival time of GBM is reduced to only 15 to 16 months. As you can also see here, GBM is the most fatal of all these other gliomas. So what we wanted to understand is what happens here. What is the mechanism post-treatment and between the recurrent GBM? So now from this whole schema, you would also appreciate that it's very difficult it's not very difficult, it's impossible to understand this mechanism because post-treatment, there are no biopsies. So tumor is gone and patient is sent home. You cannot look into the brain of the patient and see what is happening in the brain and why is there recurrence in the GBM. So how do we understand this? So for this, what we try to do is that the only way to understand this mechanism is to try to recapitulate this whole scenario of mechanism and treatment in the model systems that we will have, that we can manipulate, and we can try to understand what is happening. So for this, we took two approaches. We made a cellular model where we take the tumor from the patient samples and we make primary GBM cultures, and we give radiotherapy and chemotherapy to these tumors like you would give in a patient, and then try to understand what happens to these cells. We also made preclinical orthotopic mouse models where we label the GBM cells, and these cells are then injected into the brain of the mice. And as you can see here, because these cells are labeled with luciferase and GFP, they can be monitored in a live mice. And the tumor is developed in the mouse, and mouse is then like a patient with the tumor. And we have a handle of, what and how we want to see what is happening with this tumor. So we have these two major models which we can use and see if it can recapitulate the clinical scenario. So what we did first is to see what happens to the cellular model. So this, as you can see in this graph over here, that the U87 parent cells, when they are grown in culture, U87 parent cells or the patient samples, when they are grown in culture, we give them radiation. And we found that more than 95% of the cells are dead, just like what would happen in the patient. However, there was a small fraction of cells that we call as residual or RR cells. And we found that these cells survived. They stayed non-proliferative for about 2 weeks. And then one fine day, they start to proliferate and form huge tumors, or they filled up the whole plate. And we call these as relapse or recurrent tumors. And these cells were highly aggressive, more aggressive than the parent population, which is what also you see in the patient, that the primary GBM, the recurrent GBM, is highly aggressive than the parent population. And the same phenomenon was also seen when we grew these cells in the patient samples, or these cells as neurospheres, that initially there are spheres, then we give radiation and most of them die, but there are still few cells that survive, and these cells were capable of giving rise to the tumor again. When we put these cells in the mice brain, you can see the RR population, as you can see from this graph, is much more or highly aggressive, not only in the in vitro cell cultures, but also in the brain of the mice, that is in vivo. They were more aggressive and more tumorigenic than the parent population. Then we wanted to see if this is also true for the cells or for the preclinical mouse model that we have made. So we took these cells, we labeled these cells, and we put them in the mice. So mice at, let's say, at day 8, mouse is like a patient with a brain tumor. So we give them radiation and chemotherapy, and radiotherapy, which leads to the regression of the tumor at day 16. And by day 23, we are not able to see the tumor in the CT scan, as you can see over here. Just like in the patient, after the radiotherapy and the surgical resection, tumor is not seen in the patient. However, these tumors start to come back at day 35, and I don't have all the time points, but then these tumors were highly aggressive, and we had to sacrifice these mice. So the the tumor comes back, and it's highly aggressive, just like in the patient. Now, because this is a mice, and these cells are labeled, we have a handle to actually get these cells from the mice and try to understand what is happening over here, because this is exactly what happens in the patient. So we know that we have made a model which recapitulate exactly what happens in clinic. So it's like, what do you say? We have a handle of what is happening inside the brain of a patient. Then we can take these tumors, and because these cells are labeled, we are now able to capture these cells, which we are doing in the lab. And as you can see here, we get all these cells from the brain of the mice after the treatment or before the treatment, or any time that you would want to look at the cells of the tumor in the brain, you can capture these cells. Once we have these cells, there are multiple strategies that we are adopting to understand what what happens to these cells. But for today's talk, I'm restricting myself to DNA damage pathway and its epigenetic regulation. So, like I said, we were interested in these small amount of cells that we call as RR population that survives the radiotherapy and give rise to the relapse population. So we captured these cells from these from the cellular cultures as well as from the mice. And when we looked at them, there was a very interesting phenomenon or phenotype that we found that these cells were, we call them MNGCs, multinucleated and giant. So they were like really huge cells and they were also multinucleated. So they're called multinucleated giant cells or MNGCs. These were distributed throughout different cell cycles. So they are in the different phases of the cell cycle. They were non-apoptotic, non-proliferative, and very interestingly, they were also beta-gal positive. That is, that they had a senescent kind of a phenotype, which the beta-gal positivity is the gold standard for senescence. Other than that, since we already also did RNA-seq of these cells, we found the signature of the senescence-associated secretory proteins, that is the SASPs. So MNGCs, the multinuclear giant cells, produce a lot of SASPs, which was seen also in the RNA transcript from these cells. And also we validated some of these with the qPCR. And we know that these SASPs actually they are released in the, in the media, and they give a survival advantage to the neighboring cells and to the cell itself, like in an autocrine and a paracrine manner. So we did find that the phospho-AKT, which is survival pathway, is activated in these cells when the SASPs are there in the microenvironment. Another important thing that came to our mind was that are these multinucleated giant cells were already present in the parent population? So there is a parent population that is heterogeneous, and there are certain cells of this heterogeneous population that survive after radiotherapy. So we wanted to know if these MNGCs were already pre-existing or they were induced by radiation. So if you see over here, so after radiation therapy, when we looked at different days when these cells were non-proliferative, we found that the number of MNGCs were increasing. So if the number of MNGCs are increasing, means that it is induced by radiation. Furthermore, to confirm, what we did is we sorted the large cells, that is MNGCs, and small cells from the parent population. And we did clonogenic assays for these to see if they were more sensitive or resistant to the radiation, unlike the other cell population in this heterogeneous population. So we found that they were either more sensitive or equally sensitive to the other population, which means that MNGCs are not pre-existing in the primary, they are pre-existing, but these are not the ones that actually survive radiation therapy, right? So radiotherapy, the cells that survive radiotherapy are not the ones that were there before in the parent population. The MNGCs were induced by radiation. That's what it means. So then we wanted to understand how these MNGCs are formed. If they are induced by radiation, what is the mechanism of the formation of these MNGCs? And we did some co-culture experiments where we labeled these MNGCs green and red. And what we found, very interestingly, was that there was homotypic cell fusion that was happening within these cells, which was leading to the formation of these giant cells and also multinucleated cells. So many cells were coming together and fusing together and forming these giant multinucleated cells. So giant cells and multinucleated cells can also be formed by the other phenomena, which, because of the interest of time, I did not put all the data over here. But we negated endoplasmic, endoreduplication over here. And we found that it is the fusion events that leads to the formation of these MNGCs. This is the quantitation. Another very important aspect of this is the, I'll just go here, is the number of cell division per cell that is happening. So it's when there is a population that survives radiation, what we found that more than 80% of the population was MNGCs, but there was 10% population that looked like normal parent population. So we argued that is it possible these MNGCs would eventually die of mitotic catastrophe? and the cells that are mononucleated or that are not these giant type, they are the ones that will actually give rise to the relapse population and not the MNGCs. So for this, to address this question, what we did is we did single cell dilutions of MNGCs and mononucleated cells. And we found that MNGCs were capable of giving rise to mononucleated cells, those cells that were there in the relapse population, at a rate which was similar to the mononucleated cells, which means that MNGCs are equally responsible, in fact, more responsible, because they are more than 80% of the cells were MNGCs, in contributing to the relapse population. So relapse population is basically formed from these MNGCs only. So MNGCs are capable of proliferating and giving rise to the relapse population. Again, now this is all happening in a dish. So does it actually also correlate with the patient samples or poor prognosis of the patients? Indeed, what we found was that the samples that have that produce high number of giant cells in the culture system, they had a poor prognosis compared to the ones that had low percentage of giant cells. So it means that there is a biological relevance of this MNGCs, and it is correlated with the patient outcome. The formation of these cells is correlated with the patient outcome. Now, we know that, so like I said, we are interested to now understand, so with all these data that I had shown right now, what we did was to make sure that, yes, we are dealing with the right population. This is the right cells that we should be looking at, because these are the ones that actually survive and give rise to the relapse population. So then we tried to look at the molecular mechanism, how these cells are surviving the radiation therapy when most of the other cells in a heterogeneous population are not able to survive therapy. So this I had already shown before, that the residual cells are highly aggressive in the tumor. They form more bigger and more tumorigenic in the in vivo situation. So the first thing that came to our mind was that, is it that these cells that are surviving somehow do not get the damage itself? So the radiation is not able to induce the damage. And because there is no damage, these cells are surviving and then proliferating and giving rise to relapse population. But that was not the case. So this is the staining for gamma H2X, which is a gold standard for the double strand breaks. And you can see over here and also here that the relapse population right over here had high amount of gamma H2X, that not the relapse, the residual cells. So that means these residual cells that were there that survive after the therapy had seen radiation. It's not that they do not have DNA damage. So they do have DNA damage. And interestingly, when we did an EM of these cells, we found that they have a lot of DNA damage, and as well as their chromatin the heterochromatin is less. It is euchromatinized, which was then again confirmed by staining with the HP1 alpha, which is a heterochromatin associated protein alpha, which goes down here in the relapse population. So the chromatin of the relapse population is open. Now, this is very interesting phenomena, because the chromatin is very highly associated with the DNA damage response, because for the recruitment of DNA damage repair proteins, the chromatin should be accessible to the repair proteins, so that repair proteins can come, bind over there and repair the DNA. So we wanted to see if there is a preferential recruitment of some repair, because we know that DNA is getting repaired. I've not shown all the time points, but eventually in these residual cells, all this damage gets repaired and these cells are able to survive and come back as a relapse population. So if you would recall, so when there are double strand breaks made by radiation or chemotherapy, I've shown radiation over here because it's the system that we use. There are two major pathways by which the double strand breaks can be dealt with. One is the non-homologous end joining pathway, that is NHEJ pathway, and the other one is the HR pathway, that is the homologous recombination pathway. And Cu70 and 80 and BRCA1 and BRCA2 are the major proteins or the gold standards that are used to understand which of these pathways are activated in a cell. So we use these proteins, and we first looked at phospho-BRCA and Cu80 proteins, and we found that Cu80 was more expressed in the residual cells compared to the BRCA. What is the phospho-BRCA, which is not that much activated in the residual cells. Again, but for the DDR proteins, mostly it is not the expression, but it is the recruitment of the protein. So there should be more recruitment of the repair protein to the site of damage so that the damage can be repaired. So we did the immunofluorescence assay where we can see the recruitment of a protein to double strand breaks. And we did find that the Cu80, as shown over here, was highly recruited or preferentially recruited compared to the phospho-BRCA1, which is a HR mark, and Cu80 is the NHEJ mark. So which gives an idea that NHEJ is preferentially activated in the residual cells. However, recruitment of the protein, does it really means that the repair is happening in these cells via NHEJ pathway? For this, what we have is the HR and NHEJ repair system. These are the reporter vectors that can be introduced in a cell, and you can monitor with the GFP, you can know if the HR or the NHEJ pathway is active in the live cells. So these are all live cell reporter assays, which again showed that in the residual population compared to the parent population, non-homologous end-joining pathway was preferentially activated compared to the HR pathway. So now, like I said, that chromatin and DNA damage repair, chromatin can influence DNA damage repair pathway choices. Now we know that NHEJ repair pathway is the pathway of choice that residual cells are using for repairing their DNA. So we wanted to understand how is this preferential choice made between NHEJ and HR. So one way is by which the cells make these choices is by epigenetics, or modulating the histones of the cell, which then preferentially recruit one or the other protein. So we screened for different methylations. I'm showing here only dimethylation, but we had screened for multiple other methylations to see which of these would be activated in the residual population. And as you can see here, H3K36 dimethyl and H3K4 dimethyl were highly expressed, highly activated in the RR population compared to the parent population, while the other ones were either down-regulated, like H3K9 is down in these population, which is also a mark of heterochromatin. So it goes with the data that the chromatin is more euchromatinized. And the other marks were not, either they were not differential that much, or they were down-regulated. So then we focused on these two marks, H3K36 dimethyl and H3K4 dimethyl. And we looked at these marks in the patient population. So this RR is the residual cells from the primary culture. So we made primary cultures from 8 GBM patient samples. And we found that H3K4 and H3K36 were high, but H3K36 dimethyl was higher in almost all the samples. It was in 80% of the samples, it was higher compared to the H3K4 dimethyl. We then looked at the modifiers, that is the histone methyl transferases that can mediate these histone methylations. So when we looked at these methyl transferases, what we found was that the setmar was highly upregulated in all the RR population in the same way like the H3K36 dimethyl. And this is the histone methyl transferase that mediate H3K36 dimethyl. So it corroborates with the protein modifications. So this was the this was the transcript that was higher, the enzyme was higher, and the downstream modification was also higher in those samples where the enzyme was higher. We had, like I said, we mentioned that we had the transcript data from patient samples and the cell lines, and we looked at all the histone methyl transferase, or in fact all the histone modifiers in these, and we found that even in those samples, setmar came up as the most deregulated, or these are in the residual population from the patient samples and the cell lines. We also checked for other methyl transferases, of which only DOT1L was upregulated, but then the corresponding histone was not modified, so we did not pursue this, and we focused our attention to setmar. So setmar was the protein of interest, the protein that, or the enzyme that we thought that let's see if we knock down setmar, what happens to the cells. So we did a knockdown of setmar using shRNAs and siRNAs for the setmar. And these are conditional knockdown so that we can do knockdown specifically in the residual cells. So when we did that, we found that these residual cells that had the knockdown or that did not have SETMAR, they stayed in the state that I had mentioned before, that they were non-proliferative and they were senescent. However, when SETMAR is present, like in the parent population, they are able to come back, right? Like when you give RR population, what we had seen before is that they are non-proliferative, senescent, but they are able to overcome senescence and come back and form relapse population. However, when we knock down SETMAR, these cells stayed non-proliferative. And when we looked at their senescent phenotype, we found that they were senescent. So they stayed senescent forever. And this we kind of, monitored these cells for almost 2 months and nothing was happening. They were not coming back and they stayed senescent. And probably eventually they die of apoptosis after 2 months. So SETMAR is helping the cells to come out of this senescent state. So SETMAR is important for these cells to somehow repair their DNA and then come out. So if you do not have SETMAR, these cells are not able to fully repair their DNA and come out of the senescent state. Then we looked at the H3K36 dimethyl, because that is the protein that that is the modification that the SETMAR induce. And in the siRNA knockdown, the modification is also lost, as well as the recruitment of Q80, as you can see here in the SH1, the recruitment of Q80 is also lost. So there are two things that are happening when we abrogate SETMAR. One is that the histone modification is gone, and the other is that the Q80 recruitment is also gone. However, what we wanted to also know is that, so are these two events, that is this event where the histone modification is not there and this event where the Q80 are not there, are they connected or they are independent of each other? Like SETMAR is doing two independent things. One is that it is mediating this modification, which is now not happening, and that it is somehow also mediating the recruitment of Q80, which is now also not happening. But we did not know from this experiment if these two are connected, like H3K 36 dimethyl is connected to Q80 or not. So for this, what we did is that we made mutants of H3K36 dimethyl. So H3K36 was changed to alanine, and which we call as a mutant form. So when it is mutated, the modification cannot happen because there is no lysine at H3 at the 36 amino acid is changed from lysine to alanine. So modification should not happen. And this is exactly what we see in the mutant. There is no modification that happens. And we also confirmed this with the immunofluorescence. As you can see here in the mutant cells, there is no H3K36 dimethyl, which is shown in green over here. So there is no, in the mutants, mutation works and this mutant is not able to have that modification. Now we looked at the Q80, what is happening to Q80. And interestingly, what we found that in the mutant H3K36, there is no recruitment, there was no recruitment of Q80. So Q80 recruitment went down, as you can see over here in the red, compared to the parent population. So definitely, so which confirms that H3K36 dimethyl is required for the recruitment of Q80. So what we now know is that in the residual cells, there is upregulation of SETMAR. This SETMAR leads to the modification of H3K36 through dimethyl, which preferentially recruit Q80, which is a non-homologous end joining pathway. And the cells repair their DNA by non-homologous end joining pathway, and then they survive and form relapse population. And when the mutant cells are definitely not able to survive, as you can see over here, that these cells die of apoptosis. Unlike the SETMAR mutation, this is an interesting point because the SETMAR mutations that we had, these cells stayed senescent for very long, and then the apoptosis was induced after 2 months. However, in the case where mutant of H3K36 is there, the induction of apoptosis was much earlier, and the cells die of apoptosis rather than staying for very long in senescence and then going for apoptosis. When we put these mutants and the wild type in the brain of the mice, we see that these cells were not able to form any tumor. The mutants were not able to form any tumors in vivo as well. And then, so now we know that a non-homologous end joining pathway is doing all this, let's say, molecular biology. We tried to understand the mechanism, and what we found was that these certain molecules are involved in the non-homologous end joining pathway, and that is the pathway of the choice that the cells are making to survive. So there is a inhibitor for NHEJ repair pathway. We use that inhibitor to see, like, so if we want to take this to the clinical settings now, so we need inhibitors. shRNA and siRNA are mostly to really for our understanding to really know what kind of molecules are involved in the like doing more mechanistic studies. So the inhibitor we used, and we found that the inhibitor indeed abrogates the survival of the residual cells, not only in the cell culture, but in mice where we inject the cells. This is the parent population with DMSO and parent population with the inhibitor. And here is the residual cells from the residual cells alone, then with the DMSO in vehicle and with the treatment. And you can see that the residual cells with the treatment totally do not form the recurrent population. So there is no growth of tumor if these cells are treated with the NHEJ inhibitor in vitro or in vivo. So, so this is one part. And in another parallel study, we also found, I just wanted to show this one slide for the study because the in vivo data was very strong and very encouraging. But we also found that the proteasomal activity is high in these residual cells. And when we use an inhibitor for proteasomes in the residual cells in the brain of the mice, there is no growth of the tumor at all. So this is another very interesting target. We have this, we published it in OncoTarget with all the mechanistics, but I am just showing you the final figure, let's say, where the most important point is made that in vivo, this proteasome activity is required for the survival of the cells. And if you have the inhibitor, you do not see the growth of the recurrent GBM. So concluding, this is the second last slide. So what we did is we started with this. There is a primary tumor, surgical resection, radiotherapy, tumor is gone in patient, and it is recurring back. And this is what we wanted to understand, this what happens in between. And now using our model systems that we have, that is the cellular systems and the preclinical mouse model, what we understand now is that here where you actually do not see a tumor, but there is a residual population that is left, which are giant and mononucleated and mostly multinucleated, that secrete the SASPs and modulate their microenvironment. There is a homotypic cell fusion that happens between these cells that give them a survival advantage. The double strand break of these cells are taken care of by expression of SETMAR, which leads to the S3K dimethyl methylation, preferential recruitment of Q80, which leads to the higher repair of by NHEJ pathway, survival of the RR cells, and the recurrence of the cells. And we also found that the proteasomes are higher in these cells, and via, like I said, the whole mechanistics I've not shown, but via the activation of NF-kappa B, there is a survival of RR cells in these cells, and the recurrent GBM is formed. So, understanding all these mechanistics here, what does it tell you? It gives you a lot of window where we now know that these windows can be targeted, and that's like a window of opportunity where you can use it for therapeutics. So you can use, we used a V1 kinase inhibitor over here that leads to the abrogation of homotypic cell fusion, which again blocks the recurrence here, or recurrence of GBM. Then if you knock down SETMAR, again, you do not see the recurrence of GBM. If you use NHEJ inhibitor, which inhibits this whole process, again, you do not see a recurrent GBM. And finally, if you use a proteasome inhibitor, you do not see a recurrent GBM. So this is what we know right now. There are a lot of other studies that have added to this complexity. There are two things. It adds to the complexity. Let's say it also makes us understand more of how these cells are surviving. And apart from these windows, it gives us more opportunity for the therapeutic window, where we can use the inhibitors for getting rid of these recurrent GBM. These are all the papers that I a bit talked about. Like, there are a couple of slides from each of these papers that we have published. And now what next we are doing, this is just a glimpse of what we are doing. In the mechanistics, we have a lot of newer things that are coming up, and we are trying to understand. We have made PDOX models. So the model that I showed you, the preclinical model, was cell line based, but now we have the PDOX model ready. So all these targets that we see, or if there are more targets, that we can use the PDOX model to see what happens in the mice. Then we, like I said, that we can isolate these cells from the brain of the mice, from the PDOX as well as from the cell system. So from this mice here, we are going to isolate single cells, which we already have done and sent for single cell RNA-seq and single cell proteomics. Because of the heterogeneity, we wanted to understand how heterogeneous is this population, even though it is a small population. But then single cell RNA-seq will give us an idea like how heterogeneous this population is and how we can target these cells. And then finally, in collaboration with ICT Mumbai, we are also, some of the targets that we already have, we are trying to, to deliver them to the brain, because blood-brain barrier is another major problem in the treatment of GBM. If we can have some newer delivery methods where we can overcome this blood-brain barrier, or some newer targets that we can use in these mice. With this, I would like to acknowledge and thank all the people who have done this. But as you can see from the papers that I had listed over here, it is mostly the core E, which is Ekjot Kaur. So most of the work, she was my first PhD student. She started this project and multiple things have come out of this project. And now she is a postdoc at NII Delhi with Dr. Sagar Sen Gupta. And these are other members of the lab, postdocs and other PhD students who are now taking these things forward. Some of them are working with glioblastoma, some are working with leukemia. Our clinical collaborators, we get the samples for all our studies from these clinical collaborators. Rahul Thorat is the person who helps us in the animal facility with a lot of mice experiments. And of course, nothing can happen without the funding from all the possible funding agencies in India. And with this, I would like to thank all the listeners, and hopefully I have not taken more than what I was supposed to take. I guess I'm well in time. So thank you for patiently listening to this talk. Thank you very much. Yeah. And I'm ready to take the questions.
I can see a couple of questions, Dr. Shilpi, in the chat box. If you could address that.
Okay, wait. I'll just chat. Where do I go to chat box? Okay, here.
It will pop in down there on the screen.
Yeah, I see the chat, but somehow it's not opening. Oh, wait. I'll just stop sharing these slides. Do I need them?
You can just minimize those or stop sharing.
Yeah, I'll just stop sharing so that I can. Yeah, I think now I am a chat. All right, targeting results have failed in GBM. Which delivery system can be made to use this regarding overcoming BBS still maybe? Yes, so BB, this is like like I mentioned before, this is really a challenge, this blood-brain barrier, but we are working around it. And it's not that targeting residual cells have failed in GBM. In the past it has, but that doesn't mean that it should not be tried. And these are the cells that need to be targeted. There's no way out. I mean, if these are the cells that are responsible for relapse, you have to target these cells, but we cannot just say that this was not done before, so we don't know. With newer techniques that are coming, with newer ways of delivery, I guess this is possible, and this is what needs to be done. So we are also trying to see how we can overcome this, and it is a challenge. It's not easy. With GBM, anything is not easy, but we are trying to address that. I mean, if he wants to make some other comment, he can speak. I guess I can see him here. Then cancer stem cell survive targeting. Do we have some targeting? I don't know what really this means, but cancer stem cells are there for a lot of tumors. But I will tell you that in our system, when we look at these residual cells, there is a very small percentage of stem cells in these residual cells. And when we sort these residual cells and the stem cells, when we sort the stem cells with the so-called non-stem cells, and we put them in mice, they all have equal potential of forming tumor in a serial transplant as well. So it is not only the stem cells that are responsible, at least for GBM, I can say, because we have done the experiment, that are responsible for resistance and recurrence, but all the cells that survive with therapy, that is all the residual cells needs to be targeted, stem as well as non-stem. Do we have some targeting? Yeah, I guess I had already shown you the results of what we can target, which is because when we target SETMAR or when we target NHEJ pathway, none of the cells survive. We do not see any cell surviving. So it's killing the stem cells and the non-stem cells. And the same happens with the proteasomal inhibitor. In fact, proteasomal inhibitor, I would say it was like really clean, and it also crosses the blood-brain barrier. So it again then answers the question by Dr. Khushwant Yadav that the mice experiments that I showed with the proteasomal inhibitors, these are the experiments that are done in the mice where we inject the cells, the tumor is grown, and with the tail vein we inject the drug. No, with the oral, it's an oral, we give them the proteasomal inhibitor, it crosses blood-brain barrier, and there is no development of the tumor in these cells. So definitely there are targets. I mean, there are targets that we have, and I'm sure that other labs that are working might also have some targets. So yes, it's possible. And then, but the point is that just because you have one target or the other target, you should still not stop working. You never know when the resistance to these inhibitors or to these drugs can develop. So one has to, I guess, understand as much as possible regarding the mechanistics of how these cells are surviving. Yeah, so this nose delivery, actually, with one of our collaborators from IIT, we have this so-called nose spray. Right now, we are only trying it with the temozolomide, which is already a drug that is used in nasal spray, which is that is used in the brain. And it seems to be quite effective, at least with temozolomide. So if there, suppose we have some, see, right now we are, like I said, we are still collaborating with people. We are not into drug development, or we are not into, this industry where they change the chemicals or delivery systems, I guess would be the more appropriate word. So we have some targets, and then we are looking for the means to actually put them in the brain. What fraction of TMZ taken orally must reach the brain? I don't know. I have not done that. But what we give them is, I think, 69 mg per kg. And that is every time we give a radiation before that, like 2 hours before that, before the radiation, the mice is given. But I could be wrong, but as far as I remember, this is the dose. And I think I have it in my slides as well, one of the slides. Can we be sure? I guess, I mean, right now, I don't think right now we can actually talk, and I don't have to read. It was only when I was talking, I guess you asked them to write in the chat box, but right now we can just, you can just ask. Because every time I'm reading over here and then answering. Can we be sure that after targeting residuals, the relapse won't happen? Yes. The simple answer is yes.
Okay.
Because these are the cells that are giving rise to the tumor. And there are a very interesting, see, there are a lot of things that we have done, but I cannot show you all the data over here. So there are two ways by which the relapse can happen in GBM. Let's say, for example, after giving radiation. One is that the tumor cells that were originally in the tumor are surviving, or because radiation can also induce mutation in the neighboring cells that were not tumorigenic before, have now become tumorogenic and giving rise to relapse, right? There are only two possible scenarios. So what we have also done in the mice, right? Patient you obviously cannot do. What we have also seen in the mice is that the neighboring cells do not form the relapse tumor. Why I'm saying that is because now the cells that we have injected in the mice are the human cells, right? So we have probes for human cells and the mouse cells. And when we have the recurrent tumor in the mice, we stain them for the mouse and the human, right? And what we see, it's only the recurrent tumor is all composed of only the human cells. So no cells are coming from the mice. If the radiation was having an effect on the neighboring cells, let's say, and they are becoming tumorigenic, first of all, radiation, even if it will have an, if eventually it has an effect, we know that carcinogenesis and everything, radiation definitely has an effect, but that time period is too short to have such a recurrent what aggressive cancer cell. So the tumor cells, we know that is coming from only the residual cells. So if we target residual cells, yes, we should not have the relapse of the GBM.
Any further questions? Okay.
I have one question. Can I ask?
Yes, please.
Please.
Though the pathways that you mentioned are active in the RR cells, are they active even in the glioblastoma cells before the radiations were given, or they get activated only after undergoing a radiation?
No, so these are normal pathways. So NHEG and HR pathway are there in all the normal cells as well. So these are the basic pathways of DNA damage repair, right? But normal cells, when it gets damaged, both the pathways get activated based on what kind of cell cycle these cells are in. And there's not a huge amount of damage in the normal conditions. I would say that the cells would die of apoptosis, right? And the molecules that are involved in the residual cells compared to in the normal cells are also different. So there is a modulation of the pathway, and that's why it is very important to understand that there is a modulation of the pathway, and that's why you can specifically target only the residual cells. So this would be a residual cell specific. So it kind of, it uses the same pathway that normal cells use, but it customizes the pathway for its own benefit. So there are some small changes that are done by these cells, which are not used by the normal cells. And these are the changes that we have to understand, because these would be the molecules that you would want to target, because you don't want to just kill all the other normal cells.
Exactly.
Yeah.
Thank you.
Yes, sure.
Can I also ask a question? I'm so sorry, I was muted. Dr. Mashmi, can I please ask a very brief question?
Please ask a brief question, okay? Dr.
Yeah. So first of all, thank you, Dr. Shilpidat. I mean, thank you so much for sharing so much informative data. My only concern with this drug TMZ was that there are some papers which have got like serious concerns over its oral use, because even with the oral use, it's not able to cross the BBB. That is one.
No, but TMZ is given to the patients all the time, right?
Yes, it's the only FDA approved drug.
And actually TMZ is not our concern, because we are not dealing with TMZ.
What?
We are only dealing with radiation. We are not concerned with TMZ. TMZ has a totally different mechanism of repair. It is MGM mediated, MGMT. It is not double strand breaks. It is MGMT mediated mechanism. And it has it's an alkylating agent. It's very different mechanism, and we are not dealing with it. We are only dealing with radiation. But because in the clinic, at least in Tata, in Tata Memorial, they always give patient TMZ irrespective of the fact that they have the promoter is MGMT promoter is methylated or not, because there is no adverse effect. So basically, ideally, you should give TMZ only to the patient that have promoter methylation. But to know if the patient has promoter methylation, you have to do sequencing of all the patients. And methylation sequencing of the promoter is very expensive. And it's for the countries that are very rich. In India, it's not possible to do it for all the patients. But because it doesn't have an adverse effect, it's always given. But it doesn't, it has not shown let's say some enhanced effect of radiation or something. So you use it or you don't use it, you still see TMZ independent effect of radiation. So basically whatever is happening to the cells, unless until there are very few patients that have the MGMT, the promoter is methylated, they can kind of maybe use low doses of radiation, but otherwise it doesn't really affect the radiation dose. So radiation dose is given as a standard, and this is also given irrespective of the FGMT status. So because it's done like this in TMZ, in our mouse model, we are giving TMZ, but even if we...
Thank you so much.
Yeah, sure.
Yeah. Dr. Shilpi, I have a quick question.
Yes.
Since you are dealing with a lot of research related to radiation, right? Radiation therapy. So is there any like success story, and you are since associated with ACTREC and TMC. So is there any story where you have been able to take such positive results in patients at ACTREC or in TMC?
No. So right now what we have, the studies that we have from our mouse model is preclinical. And now we will be taking it, see, changing anything in clinical practice takes forever.
Yeah.
We have such good targets. We have mouse model to really show that, how beautifully it works in mice. But then we have to do multiple, even with these doses, with the radiation dose, the dose that you give to the mice and the dose that you have to give actually to the patient would be different. So it's a long path, but we are getting over there. So now that we have this inhibitor for NHEJ pathway, and we can also do clinical trials in TMC, which gives us a benefit of, what we are doing in on bench to take it to the really clinics. So we are right now in that phase, but it's going to take some time. But I guess in coming years, you will hear that success story of, bench to bed site.
Since your presentation was, or your research work is highly equipped with, single cell proteomics type of study, targeting SETMAR, and also I really see how it can be taken really to ACTREC and TMC. So these are highly equipped phenomenon which you have used in your work, and it's highly impressive. Dr. Mayur would want to add something here, sir?
I am in observation mode because I am going to be collaborating. So
she's your prospective collaborator. You can
yes. I am looking for people who are really have the same interest and willing to take the risk.
So because the whole workshop is based on your kind of work, so naturally we will be reaching you soon.
Sure.
So I must say that you have really given a good start to our workshop with a stimulating presentation, which was equipped with right from your lab studies to CT scan. So it was really, we enjoyed it, and it was a delightful, session for all of us. And I would really like to thank you again for accepting this assignment and volunteering your time for this activity, educational activity, and also for the growth and enhancement of the skills of the researchers in academic setup. So thanks once again.
The pleasure is all mine, I would say. I mean, it's always nice to interact with people who are really interested to listen to us. Scientists are always wanting to show off their data. So it was really
with those hopes, I think we will be touching future as well. Thank you so much.
Yeah, thank you. Thank you.
Thank you, Dr.
Thank you, Mayur.
Thank you.
Bye.
We are quite right in time, people. So as Dr. Shilpi has set the platform for DDR, DNA damage response, we have our next speaker who has a quite enriched domain, who has been working in IIT Gandhinagar. So do we have Dr. Shivpriya with us?
Yeah.
Hello. Good
Very
morning.
Good morning. Good morning. I don't know whether Dr. Shilpi left because we overlapped at MIT when I was a postdoc and she was also postdoc there. So I just wanted to say hello, but I think she left. It's okay.
But I found a link between your talk and the platform which she has set in her first lecture.
Oh, okay. No, I know her personally also. So I just wanted to say hi, but I think she left. Thank you. So shall I share the screen?
I'll just give a quick introduction about you to the participants. The DNA damage response, comprising DNA repair and cell cycle checkpoint pathways, is an attractive target for cancer therapy. The collective DDR offers a rich source of anti-cancer drug targets. Our next speaker for the day is Dr. Shivpriya Kirubakaran. She is working as an associate professor at Cancer Chemical Biology at IIT Gandhinagar. Her area of research is the discovery and development of selective cancer therapeutics. Her focus is cancer-related protein kinases, DNA damage response pathways, and novel targets for anti-cancer therapy. She has also been working on designing of small molecule inhibitors for the proteins for anti-cancer drug development. She has published about 100 papers in scientific journals such as Journal of American Chemical Society, Journal of Medicinal Chemistry, Cancer Research, and many more. With such quick introduction, I would like to invite Dr. Shivpriya to share her screen and take us to the journey of targeting DDR kinases, new age cancer therapeutics.
Thank you, Dr. Moushmi. It's always pleasure to talk to people who actually appreciate the other part of the research, which is the application part of the research. And I'm very thankful for this opportunity. And I see Kapil here. So my whole lab is entirely actually started by Kapil, Dr. Kapil now, your own faculty. So I'm very happy to see him and then present this work. So since I'm presenting to your institute, I'm not presenting what Kapil has sparked in our lab, and then that work is going on really well. I'm very proud of that work that is going on. But today I'm going to talk about some. So I was hearing last 15 minutes of Dr. Shilpi's talk, where there were a lot of questions about success stories. So I was thinking, oh my God, I should not talk after Dr. Shilpi, because it was a wonderful talk, and I missed the talk because of other clashing meetings. So what I'm going to show here to all of you is probably the lot of failure stories of my lab. But eventually I'll make everybody happy. Okay, so I don't want to tell that. I will only tell you the failures of my lab, because all the failures what we have been looking for this last few years has been really helpful to make us very successful. Okay, so I think my am I sharing the right slide? I'm just checking once again. Okay, so because the title is a little bit changed, so I'm not sure. Okay. So our lab is about 7 years old at IIT Gandhinagar. So we have started working on the drug discovery part of cancer therapeutics. But what we understood in the last 7 years is that's very important to also study the mechanistic part of these drug molecules. So if we know the mechanism of how this molecule works, it would be much more better for us to understand eventually how the molecule could be modified, or if it is even clinically fails, what are the things that we could potentially do to make that molecule more successful. So we started about 7 years ago at IIT Gandhinagar. So just to give a brief introduction, maybe for this audience, this might not be very new introduction, because you are working in the pharmacy area, and then you probably know much more better history of cancer. So, but just give a brief introduction. Cancer is not a very new disease. It's the first documented case of cancer is in 1500 BC. Okay, as you can see here. So it's been there for a very long time. And this is one of the pictures that is actually documented, where the tumor has been removed by surgery in 1600 itself. So it is not that cancer is very new to us or to human society. But right now, where are we is 1 in 3 people will develop cancer during their lifetime. As you can see here, this is not my data, this is WHO data. So 1 in 9 people will have the risk of developing cancer if they survive for the first time. 2 in 9 people will have the increased risk of developing a second primary tumor. What does it mean is that if somebody has had a surgery, had enough of chemotherapy, and looks like that cancer is completely gone, there is always a risk of developing a different tumor type. Like somebody has a breast cancer, but after 20 years, the same person may develop a skin cancer. So the second primary cancer is also a big risk. So it doesn't mean that it's uncurable. There are very successful stories where people haven't got cancer after the first treatment has gone very successfully, even after 20-25 years. Okay, so, but if you see the incidences of the cancer, new cancers worldwide, it is actually almost double between 2002 and it keeps on increasing. Okay, and I call cancer and infectious disease as a deadly duo because the current trend continues, cancer would become the leading cause of death worldwide. Okay? This is from the AACR annual reports. And quickly, if you want to check how this world cancer scenario, where it is more of a spread. So this slide shows that geography of cancer. As you can see here, it is not only spread in only one country, 2 countries, 3 countries. Across the world, where the most of the places the cancer is reported. It's not that only India has these many number of cancers and only Japan has or only Russia has. As you can see here also, the number of different types of cancer, which is also spread overall, is really high. And when you go closer and see the geography of cancer in India, as you can see here, this is in a public health, Institute of Public Health has published in Indian Journal of Public Health, where you can see they have analyzed state-wise of the cancer incidences or different cancer type that would probably be more prevalence in particular area. So as you can see here, we live in Gujarat, right? Right now, IIT Gandhinagar. So in Gujarat, what is the most most higher cancer patients are there in the neck and oral cancer? Because a lot of tobacco is used in this part of the country. So like that, it has several different reasons, or the environmental factor also is very, very important. Where you live, which part of the country you live, what is the environment of that place. So sometimes it is also skin cancer is coming really up in this part of the country because the UV radiation in summer is really high here. And it's a very costly disease, right? So cancer, it is not definitely affordable by very common person. So why is because the research that has been going on in cancer from last maybe 100 years, 120 years, 140 years. But if you see the recent reports, the incidences that happened in cancer is from 2012 to 2035. It is going to be 24 million people would be affected, or the cancer incidences that we say would be in millions. But the amount of money that is actually put to do this cancer research is going to also be increased to almost $500 billion into 2030. So 2030 is not very far away from today. And this also makes the FDA to approve cancer-related drugs much more faster than the regular drug approval. You see, in last 20-30 years of the approval from FDA, the hematology and oncology are the ones which actually takes up the chart much higher level than any other thing, and then followed by the infectious disease. That's why in the beginning itself I call infectious disease as well as cancer as a deadly duo. So if you see the cell, I call the cell as a big bowl of soup, because the cell has like a bowl of soup has water, it has vegetables, it has sometimes meat, it has all sorts of things, right? Lentils. So it is actually a complete meal that we probably would call. So similarly, a cell is also like that. It has several organelles, as you can see here, but it's a very complex system. So how are we actually going to see into the cell? As a chemist, I'm a trained organic chemist. So when I see this picture of the cell itself, I'm very confused. Okay, so what are the major things that happens in the cell that could be major reasons for this cancer? Either it's a starting of the cancer, or a proliferation of cancer, or even finally the apoptosis of the cancer cell. So we don't know whether what particular signaling pathway or what particular protein is going to be more active in this whole complete bowl of cells. So let's see how to balance them. So this is the most important slide. I would say that there is a very huge balance between apoptosis, which is the cell death, programmed cell death, and the cell growth. If the cell division and the cell death, it doesn't have an equilibrium, then we will have problems. So if there is a more cell division, okay, and then the cell death is not happening, then you have cancer. Okay, so if you have less cell division, or the vice versa. So if there is no proper balance, then we are not healthy. So how a normal cell actually becomes a cancer cell? So there are a lot of reports that how a normal cell would become a cancer cell, and there are a lot of reports and a lot of literature has already been given. This is one of the major picture from the hallmark of cancers. These are the hallmarks of cancer, which is now there are 7 hallmarks of cancer. This is only 6 hallmarks of cancer I've given. But when you see, these are the major important hallmarks that I can just figure out. So one is that the genetic changes that happen in the cell, which is important to understand. This could be because of the inactivation of tumor suppressor genes and activation of proto-oncogenes, and also inhibition of apoptotic machinery. So all these terms would be like French and Greek if somebody doesn't know biology. But I'm going to simplify these terms with chemistry. So so I'll just skip this. So if you see the major, so all those previous complicated words in biology I told you is mostly maintained by, there are something called receptors, proteins, DNA. So whatever the part of the cell, right? So now if you want to go one step ahead and then see what are the major targets within a cell that can be therapeutically viable. So the first one is mostly targeting the receptors which are on the cell, which also part of tyrosine kinase family, like the receptor of EGFR, HER2 or something like that. These are some examples. Are targeting something called signaling proteins, where the protein kinases comes, or angiogenesis inhibitors are targeting DNA replication enzymes. So these are some of the very well-known targets. But why we have chosen protein kinase as a drug target is that the kinome is a very vast target potential. It has about 520 kinases. So these are the, if you see the market or the FDA approval, these are some kinase inhibitors which are already in the market for several cancer treatment. As you can see here, this is a kinome tree that we call. So each of these bulbs or the blobs that I call, so if you see just the yellow one, there are several yellow ones. That means a single molecule or single drug of one of these, the drugs that I have shown here, can actually go and bind to several different kinases. So why? Because the kinase inhibitors are, we don't know whether they are structurally similar, structurally different, but since the kinase binding pocket is almost similar for most of the kinases, either it is ATP binding pocket or GTP binding pocket. And if a drug is actually made based on those based on the binding pocket, then there is the selectivity issue comes. Okay, so that's why if I show this kinase inhibitors and then showed the previous slide, the one inhibitor can go and bind to different kinases. So that's a major challenge in kinase drug discovery. So just quick introduction of protein kinase. It's a very simple chemistry reaction. What a protein kinase does is that there is if there is a signal that happens inside the cell, a protein kinase adds a phosphate. It's a simple reaction, but doing the simple reaction in our RB in chemistry lab is extremely difficult. Okay, so it is not easy that you add a phosphate salt, you take ADP, and then just ask put up the reaction or heat the reaction or do whatever it is, but it's not going to happen. So in chemically, this particular single modification is very difficult, but protein kinase does this reaction so fast. Okay, and then protein phosphatase is the one which actually removes this phosphate. So this is a very big signaling cascade inside the cell. When the signal is on, then the protein kinase is active. When the signal is off, then protein kinase is inactive. So this conformational changes that actually modifies the kinase activity in the protein-protein interaction, and it's a very big signaling cascade. Normally this signaling cascade of kinases is compared with the New York subway, because the New York subway is so complicated. If one train stops even for a few seconds, then the entire subway will be jammed. It is exactly like that. For one signal of this protein kinase stops. So because this protein kinase comes from top, that is means that upregulation to nucleus. or to DNA. So wherever it stops, then the entire down regulation of the entire signaling cascade will directly stop. So this is very important in cell division as well as in several other cell functions. So there are two major types of kinases. One is serine threonine kinases, other one is tyrosine kinases. So that depends on which amino acid that particular kinase phosphorylates. So as I told you, the protein kinase, if you look at the structure very closely, it has a ATP binding pocket. As you can see here, there's an N lobe, there is a C lobe. This is called the hinge region. If you go more closer and look into the protein kinase ATP binding pocket, as you can see here, there is a nice hydrophobic region, there is a hydrophilic region, and there is a nice binding pocket of ATP. Once this ATP binding pocket is actually stopped or mimicked by another inhibitor, a small chemical molecule, so then that complete, this is the first thing that any chemist or anybody would think, right? So that you make a molecule which is very in competition with ATP, but the concentration of ATP in our body, in our cell, is really high concentration. So making something very competitive to ATP would also have a lot of implications in our biological system. So this is a basic idea of many kinase inhibitors. But if you see the milestone of our journey of kinase drug discovery, it started way long 100 years before, but all of them has actually started becoming really, really important, and really people started working on kinase drug discovery only after we start getting several kinase X-ray structures. So if you see that there are type of binding that each kinase had, there are allosteric sites of the kinases which were discovered because of X-ray crystal structure. And then there is a huge blockbuster drug Gleevec came into the market. So these are the types of inhibitors that were actually made to bind to kinases, which were not only competitive inhibitors, but they also had allosteric inhibition. So if you see this entire history of drug discovery for kinases, it's been extremely successful. So that is why it was also very interesting for us when we started when I started my lab. So our interest is that we wanted to see when you want to target the kinase. So we all had enormous of literature that why kinases should be targeted and what part of the kinases should be targeted. But what are the kinases that we can target? You know, there are several kinases which has been already very well studied. So we wanted to take up a kinase which is very important in DNA damage and repair. So when a chemotherapy or a radiotherapy is given to a patient, the major thing that happens in the cell is the DNA damage. So the DNA damage can happen as in a single strand DNA damage or a double strand DNA damage, depends on what is the therapy that is given. So once the damage happens, there are these two major master kinases called ATM and ATR, which comes and repair the DNA. So once, even after giving the patient this treatment, since this repair mechanism is also very highly possible in cancer cells, the cells were actually repaired. So the cells were not getting died, or instead of that, they were getting more mutated because of all this modifications that can happen in DNA. So we wanted to see whether this ATM and ATR, which are master regulators, can be targeted. And another interesting point of ATR and ATM, they are also very well overexpressed. They are overexpressed in cancer cells, and some of the cancers had the survival mechanisms because of this, having this ATR and ATM. So if you see, there's as I told you in the previous slide, when the DNA damage happens because of IR radiation or UV radiation, you can see the double strand breakage happens and a single strand breakage also happens. So this eventually will take care of cell cycle arrest, apoptosis, and also DNA repair if this particular repair is done by ATR or this ATM. So ATM has another kinase which is phosphorylated by ATM, is called CHEK2, and then ATR has another kinase which is called CHEK1, which is phosphorylated by ATR. So these two kinases are also very, very important in DNA damage, and then these are all very well studied pathway, but there are not particular specific inhibitors for these pathways. So we were very interested to see whether these pathway can be inhibited, because ATR inhibition, it's also important for the cell survival. So if you see normal cell and cancer cells, so in the normal cell, there is this G1 pathway and there is the SG2 pathway. So even if ATR is inhibited in normal cells, the normal cells will use this pathway to become to be survived, and then it will go on. But if you have we don't have this particular pathway, which is the SG2 pathway, in case of cancer cells. So the cancer cells only depend on this ATR pathway. So if the ATR is inhibited, then there will be a incomplete replication and then accumulation of premature chromatic condensation, and then the cells can actually go to death. So this is something called that ATR inhibition, and whether if you inhibit ATR in cancer cells, will the cell survive or not? There are a lot of reports on this particular mechanism. So that is also another reason that we chose ATR. So these are the known inhibitors of ATR and ATM, but all of them had some difficulty in case of, okay, so that's what I said that I missed this. Okay, just a minute. Just give me a second, I'll just. So these inhibitors of ATR, ATM, most of them are reported by AstraZeneca. So these are all reported in 2010 or 2012 in that time. So that's the time I was actually finishing my postdoc at MIT and Harvard. So where I was very interested to take forward these inhibitors and see whether these inhibitors can we make a ATR specific inhibitors? But all of these inhibitors actually failed in clinical trial in last 6-7 years just because of their selectivity. So when I started the lab, then we had one lead molecule which inhibited mTOR, which is also a master regulator in cancer. And then, as you can see here, this is something called a taurine-2. This is a specific, we wanted to make a specific, when I was working with David Sabatini lab, then the specific, there they work on mTOR. So there was a inhibitor that was actually made, it's called taurine-2. But in DiscoverX screening, we also found that this actually hits ATR and ATM also. So we wanted to see whether this molecule can be taken as a lead. And then when I started the lab, then my PhD student Altaf Sheikh started making this generation 1 and generation 2 inhibitors. This is almost 7-8 years old work, because he was my first student and he started actually making the in-house inhibitors, which had 2 different scaffolds. One was resembling more of a taurine-2 scaffold. The other one is resembling a different scaffold, as you can see here. So we made, right now we have about 120 molecules. So at that time, we also had this Schrödinger suit so that we can model these ATR inhibitors and then see whether these inhibitors can be potential inhibitors for only ATR. But another challenge was that there is no ATR crystal structure. So we took the structure from mTOR, which was solved very recently, not crystal structure, but the cryo-EM structure. And then we modeled ATR kinase here. And as you can see here, the molecules that we were thinking that will have a better potentiality actually showed a very good binding through docking. So we did for almost 120 molecules. So I'm not presenting all the data. These are all kind of published recently. So the synthetic schemes are very it might look very straightforward, but there are a lot of optimization that Altaf has to do. So we made these molecules. We actually wanted to study these molecules because we also, as a pharmacy background people, you also see normally a lot of anti-cancer, antiviral, antibacterial, right? We really don't really go and study mechanistically whether they are really anti-cancer, whether they are really antiviral. So we wanted to, after making the molecules, we wanted to see a quick check on the cell viability, which we usually do it by cell titer glow assay. And then we got some kind of a GA50 value, which would be really nice to compare with the taurine-2. As you can see here, the 2 molecules, which we call it hit molecules, were almost comparable with the taurine-2. So they were in the nanomolar range. So we wanted to take it further and see whether they are very specific only to ATR, ATM, and mTOR. As you can see here, taurine-2 The phospholipid check 1, as I told, it's for ATR, and this is for mTOR. And then SPK98 is the molecule that we are studying at present. So this molecule is also a dual inhibitor for mTOR as well as ATR. But what we wanted to learn is that if it is inhibiting ATR in the cellular level, whether we can microscopically capture that. So we went ahead and then see whether the inhibition and localization in phosphorus check 1 antibodies to see whether this molecule is going to inhibiting this phosphorylation of check 1, which is a readout for ATR inhibition. As you can see here, this is not very specifically inhibiting phosphor check 2. That means it's not very specific to ATM. So it is more specific to ATR, and also it's quite potent with the mTOR also. But we didn't have those antibodies to study for mTOR, but that studies are undergoing right now. Another important thing that we could able to find out is that the premature chromatin condensation, PCC, what we call, is an important step in ATR inhibition. So if ATR is inhibited, then the cells would be going through this something called premature chromatin condensation. As you see here, the molecules were inhibiting, especially the SPK98, is inhibiting ATR very specifically. That's why we have PCC here. But taurine 2 is actually not inhibiting. So you can see a normal mitosis. This is a very interesting study. That this study is, though the preliminary study we published already, but we are actually in the manuscript is in the review for many other many microscopic microscopically important mechanisms that we could able to solve. So that paper is in review. So hopefully when I talk to you next time, we will be able to show you more interesting stuff on that. So this is what I said, no? So when we wanted to study a molecule, a single molecule, we just chose one molecule out of 150 molecule, and then want to see how exactly this molecule, so how far we can go and see, and then solve the mechanism. That is what we were intended to do. As I told, that ATR structure was a challenge because it's a very big kinase, 300 kilodalton kinase, and it also has a lot of different domains, and ATR, all these domains are important for the kinase to be active. So, but we wanted to see whether this without this FATC domain and the 10-terminal domain, whether we can clone only the kinase. So, along with Altaf, my first student in biology, he was Rashmi, who was also joined at the same time. So, Rashmi started working on cloning of this ATR kinase domain. So, as you can see here, we had some good cloning strategy. And then we started cloning ATR kinase domain and also ATR full length domain. But almost 26 times she failed. So almost for first 3 years, she had only the cloning of ATR in her presentations, where she would show the slides and say that this is not working, this is not working, this is not working. That was quite a very challenging time. I would say that it's a challenging time for me, but it's a very frustrating time for Rashmi. But Rashmi is an exceptional student of biology, so she didn't give up. Okay, so what she did is, finally she was able to get this purified ATR, human ATR, in MBP tagged vector. So as you can see here, there's a very pure ATR protein we have got, and we have also started doing all the assays. And then I'm very, very proud that I could publish as an organic chemist, I could able to publish a paper in biochemistry, which was also nominated as a very fine paper in biochemistry. So by reviewing this paper, Professor Michael Shedman from NIH has become a very big collaborator with us to work on ATR. And right now Altaf is a postdoc with him. So this is what I would call that is after so much of failure, the first paper when it publishes in biochemistry, I think we were very, very much thrilled, and I'm sure it would have been a great learning experience for Altaf and Rashmi. But that is what I would also say to the students, that it's fine to have failures, because there is no positive or negative results. The results are results. So we have to take up the results in such a way that what we understand from those failures, which we implemented immediately in another kinase, which I will just show you right now. So when we were having so much of failure and so much of frustration, so much of difficulty in working on this kinase, so we had another kinase which was just published. It's called TLK, tosyl-like kinases, which is also very important in DNA repair mechanism. But there were no inhibitors reported for this kinases. So what we thought is that we can use all the strategies that we are using for ATR and see whether we can clone this kinase. So because we had all the failure strategies, there is another PhD student who actually cloned this kinase. 1 minute, please. I told you I'm in a meeting, please don't call me. Yeah. So I'm sorry, I'm also Dean of Students, so I get very frank calls sometimes. I have to pick up and then please, I'm sorry about it. So there is a high yield of expression and purification of TLK1B in E. coli. Normally, it's very difficult to have a protein which is mammalian expressed protein, which we carry it to express in E. coli. But because of those ATR projects that we had, we could able to construct and design a vector which is called a pet duet vector. I'm very thankful to Professor Vijay Tiruvengata because he came up with this wonderful smart vector we call. And there is a biology student who joined as a second student of mine, Siddhant. So he started working on this TLK kinases, and he within short span of time he could able to clone this kinase. As you can see here, we got almost 10 milligram per ml of the protein, of the pure protein. So since this expression studies as well as all this bioactivity of TLK1B is very new, we could able to publish this in scientific report within like 30 days. This paper got accepted, but we have submitted this to Nature Chemical Biology and then they directly sent it to scientific report. That's what normally happens when you have to publish your first papers. So we were very happy that we were able to get this kinase very quickly. So what we did is that we wanted to check whether we can design small molecules for this kinase also, because we have a kinase, we have a set of molecules that we can actually talk about more as an ATR inhibitor. So we wanted to see whether is there initial screening that can happen, that can actually happen with our molecules, as well as some new molecules can also be screened for this kinase. So staurosporine is a pan kinase inhibitor, and as you can see here, this is one of the THD is one of the initial hits for TLK1B. This was published by Sharon's lab in 2014. So we took these two molecules and then see whether we can design some new molecules that can be potential TLK inhibitors. So we also had some literature to see what are the portions that we don't want to have it in this particular inhibitor, and what are the portions from staurosporine we want to introduce. So we came with a set of molecules. As you can see here, this is synthesized by another PhD student called Javina, who is a chemist, who is also recently graduated, and then she is in University of California now. So we had the protein in hand, so we had the molecules, and then we actually started developing an assay for this molecule. So you can see here J54 and J56 were the molecules which were inhibiting this particular kinase TLK in a very good activity. So we wanted to take this molecules further and then see whether we can take this molecule to any further level. So we did all the assays, cytotoxicity assay in different cancer cell lines and colony formation. So this is the point where Siddhant got Fulbright Fellowship, and the person who is a pioneer, Professor Benedetti from Louisiana State University, he's the one who hosted Siddhant for this project. So Siddhant went there and then he actually did enormous work on this particular molecule, two molecules on prostate cancer cell line, because he was Professor Benedetti was very interested to study TLK protein in prostate cancer. So this was a really golden opportunity for us, that we have a molecule which inhibits TLK. Is this molecule can be taken into clinical trials, preclinical trials at least. So we did the cell, all the cellular assays in his lab, including all the cytotoxicity assay, everything. And also we screened this molecule, particular molecule in different cancer centers. So Siddhant spent about 11 months. So that also helped us to take this molecule to animal model. As you can see here, quickly, I will just show you the final slide. This is the control, and this is something called bicalarmide, so which is a known prostate cancer drug. So this is our molecule called J54. So when we have the combination of J54 and bicalarmide, then you can see that the prostate cancer is completely reduced. This happened in 50 days in mice model. So we are extremely excited to see this. And then the preliminary studies also as published in Cell Press recently. So what we have understood from this studies is that we need to have we need to have a clear idea of how this molecule actually mechanistically works. So it was very important because Professor Benedetti is a biologist and then I'm a chemist. So I was thinking that there might be a lot of miscommunication in what we want to do, what he wants to do, but nothing that those kind of sort of things happen. That's because we had a student from our lab going to his lab, because that student helped us an anchor to complete these studies. So right now these molecules are in preclinical trials, and we have also studied PK/PD of these molecules. So I cannot disclose all those results now, but I will be very happy to talk to the same about the same work in future. So in summary, what I would really like to tell you as a take-home message is that for the first time, the equal expression of these mammalian kinases, kinase domain as well as the full-length domain has been done in our lab. So right now we have almost about 180 molecules, which we normally screen as an in-house screening for high-throughput screening for kinases. So we also have 6 different kinases in our lab to study. So most of them are actually involved in DNA damage and repair. So some of the molecules, as I've shown here, SPK98 shows very selective inhibition towards ATR. So we want to take this molecule also further. So even for that molecule, microsomal studies, preliminary PK/PD studies we have already done. So we are hoping that we'll take that molecule to animal model now. So we have been able to synthesize a specific small molecule inhibitor for TLK, and then this can be a very good preclinical and clinical candidate for prostate cancer. That is a work which is going on in progress. So as I told you, that we also have a panel of kinases for small molecules to study any high throughput screening. So these some of this work is patented in India as well as in the United States, which also gives us a lot of opportunity to license these molecules and then study much more efficiently. I would completely dedicate this complete work as a collaborative effort between synthetic organic chemist of my lab, biochemist of my lab, protein crystallography, as well as molecular biology from Professor Vijay's lab, and cell biology, and we also started working with some clinicians. So as I always end up in some of my talks, that even in 1902 in Nobel lecture, Professor Emil Fischer has already told that the progress of science cannot be by one person, but it will be a combined effort of brilliant people who works together. Okay, so it can only be a planned collaboration. So I also always put this as a slide for students. I don't know whether the students are there in this talk, but unlocking future is very, very difficult. Okay, so we normally dream about future, what we want to do. Particularly in my lab, our dream is to bring affordable medicines to cancer, and that drug has to be discovered in India. So that is my ambition, goal, whatever I say. But what we are actually looking is that we are looking at small things, which are the small molecules. As you can see in this figure, it is from a movie, Ben Affleck movie called Paycheck. So where he unlocks the future with all the small things. As you can see here, a key, there is a crossword puzzle, there is a pin, there is a spray, but he uses all of this in one or other way. So we also here to use all the small molecules that is actually present in the universe. So we call in a chemical universe 900 million small molecules are there, but we don't have a proper, or we don't have a specific drug for cancer. So so I think we are following our dreams, and we really want to unlock the future with the small molecules. And then, as I told, it's a important interdisciplinary at every level is very important. Otherwise, it would be very difficult to do anything by myself. Organic chemistry, biochemistry, molecular biology, structural biology, cell biology, and so on. So I also acknowledge the funding agencies to support this work, because it's impossible to do cancer drug discovery with very small amount of money, or with whatever is available in the university or the institute. So I'm thankful to IIT Gandhinagar, SERB, and FIST for their grants, and also extremely sincerely thankful to Ramanujan Fellowship, which helped me to build up my lab. Which was very, very important, and Fulbright Fellowship for Siddharth as well as to me to work in Professor Beneditti's lab, DRDO and DNDA that we have been collaborating for a long time, and Government of Gujarat for biomedical research funding. Our collaborators, Professor Vijay Tiruvengidam, Professor Benedetti, Professor Michael Shidman, and Professor Richard Bryce, who are all have been extremely nice and extremely very much grateful. And they are the best collaborators that, and then I miss Kapil's name here, but so another occasion, I think I will present what we are working together. So our group is really big, and you can see here many of my first students. I'm very grateful to Altaf and Rashmi, along with Kapil. They started the lab from the first buying from whatever they bought from Kim Weiss. They only brought from the first RB. Both of them have very well settled in United States and doing really well. Siddhant and Javina also graduated now, and both of them are also in United States. So I'm very grateful to this first few students, because without them, my lab wouldn't have been where it is now. So the most important is to do interdisciplinary science. As you can see here, these biology students are always very happy because they are very smart working biology students. But you see that most of my chemistry students are always tired because they are the hard working chemistry students. So you can see Kapil a few years back how he was. Altaf is here, Rashmi is here, Siddhant is here, Javina is here. So I'm very grateful to this first four students of my lab. So lab is going on very well. This is my current group after everybody left. And this is our campus, which is built from the scratch when 2013 when I joined. I think the previous photograph is stated when we did the Graha Pravesam of our lab in 2016. So this is how IIT Gandhinagar looks. So I welcome all of you to visit the campus once the pandemic is over. And thank you so much for the opportunity. I hope I did something which is interesting. Thank you so much.
We'll take few questions if you are.
Yes, please. Yes.
Dr. Jaya had texted something related to E. coli, where you had mentioned. She has mentioned E. coli can be explored. I guess there was a slide where you have used E. coli for expression system. Maybe something related to that. The question is not much clear.
Yeah. So that's what we are not actually using mammalian expression. We are using E. coli expression only because I think this is from the previous, some of the questions are from previous, I think Shilpi's talk. So yeah, Dr. Jaya, whatever systems we are using is only E. coli expression. We are not using any mammalian system for now. Yes. So it can also, so we are, I think Ms. Kalyani is asking this question. So eventually we want to use this kinase inhibitor as a single chemotherapeutic agent, but along with radiation therapy if they are given, because since we are trying to target ATR and ATM, so along with radiation therapy, if they can be given as an adjunct therapy, that would be, I think that would be much useful. No toxicity to normal cells. We have also studied, but I haven't shown those slides here.
Are there any further questions from participants?
Yeah, so madam, it was the presentation was too good. I think so. You started with so it was like a story. Okay, say let me ask you. I think you have shown the say epidemiology of this cancer, so across the different states, and definitely there were some states where you have not shown any such occurrences of cancer. So what may be the reason for that?
Dr. Satish, I probably shown almost all the states has the incidences of cancer because it might be the percentage. So that is probably not very clear in that. But there is no state that in India that we don't have cancer.
But in South, you've shown that there is maybe because of spices also there is a possibility of cancer. But I heard the spice, even rasam is one such ingredient which actually anti-cancer. Yeah.
Yes, because we use a lot of black pepper and other spices, which is actually has a lot of antioxidants and many other the food related things are very important. If you see Northeast, if you see very Northeast like Meghalaya or just way up Northeast, the Tripura, so those places incidence of cancer is very less. They say that because the environment is very clean and people but if you see not bigger cities, even Ahmedabad, the oral cancer is the highest in Ahmedabad. Okay, so but it is not as much as high. It's probably in either Kutch or somewhere. So though everything is in Gujarat, so the number of incidences also depends on the city where they live.
Yeah. So thank you, madam.
Thank you.
Do we have any further questions from the participants? Dr. Shilpi, I must say that I can see the collaborative part between you and Kapil, that how you have been using this, and you also showed young Kapil in your lab. So that was the most interesting part towards the end of the presentation. I really like that study where you have actually used that ATR kinase docking studies to say, those are the type of work we are also doing at SPTM. I think Kapil would also agree. A lot of docking studies, and then we are trying to create those inhibitors and trying to do. And also the part where you have showed that small molecule inhibitors where you have a panel of kinases. Yes. Can you just throw a little more light on that, where you are using panel of assays, where you have commented something like that?
Yeah, so there is a company which is very expensive. So they have all these important kinases, and then they do something called kinase assay. Okay, so this is a very simple assay, which is a kit available, but we don't have all the kinases. So that is the biggest challenge. So the kinases are not available in our country. So the kinases are available with only Discover-X. So when we send one molecule, they will screen against all the 500 kinases. So but they don't have ATR and ATM right now. Oh. So they have only the kinases which are maybe 420 kinases they have. So the 100 TLK also they don't have. They recently added TLK and TTK and those kinases, and we are only supplying this TLK now to them. So what we have in our lab is 6 kinases which we expressed the kinase domain as well as the full kinase. So sometimes only the kinase domain is active, sometimes they are not active. So in case of ATR, both kinase domain as well as the full length kinase is active. But all those kinases are with the tag. Sometimes the tag also matters a lot in the conformation of a kinase. So when there is a MBP tag, it is a maltose binding protein, which we use as the tag to purify from E. coli. So these tags, when we remove the tags, the kinase will not be active. Again, the kinase will fold in, the kinase domain will fold in different way. So with a lot of efforts in last 7 years, we have 6 kinases in the lab. So the molecules, whatever we synthesize, we normally run through this panel of kinases.
Yeah, because when you talk about these tags, and what comes to my mind when you're talking about kinases, a lot of involvement of cofactors
yes.
when you are tagged. So have you observed any such type of effects of cofactors in your studies when you are dealing with such active targets which are kinases?
Yeah, so recently there is a kinase called DNK. Where Professor Vijay Venkatam and his lab, his student has developed a real methodology, and they were working on something else, and they had this kinase as one of the cofactor. So when they were doing this pull down assays and then studying, so they also published recently in ACS, and reviewer of that paper has actually offered this student as a postdoc. So this is another part of the lab which is actually expanding, because I am not a biologist. I am more interested in how my molecule works on that. But he is more interested, Vijay's lab is more interested in figuring out the cofactors and figuring out whether some chaperones can be actually participating in this kinase signaling and all those things. Yeah.
If we don't have any further question, I would like to thank you for taking so much time out. I know that you are extremely busy and going on with really serious stuff out there, which Kapil has briefed us. So I really thank you once again.
No, I think my talk was, I don't know whether I did my talk justification to the talk, because there was just so much yesterday till 2 o'clock. We were all having some meetings. So I
think what researchers we must have picked few key points from each of the presentation. That is the aim, that it has to go with the long-term development. So it's not going to be so tricky always, but yeah, some few important key points are always we are going to keep with us. So I'm sure it is helping us in that manner. And once again, on behalf of SPTM, I am offering you a sincere appreciation to join us and contribute in such with such an excellent scientific presentation to the workshop. And see you once again in future sometime. Thanks once again.
Thank you. I would be glad to visit you soon. Yeah, thank you. Thank you, Kapil.
Thank you, Dr.
Thank you. Thank you, ma'am. Thank you.
I think these were two presentations, these were two lectures which were planned for the morning session. So to all the participants, I would request that we can break for lunch now, and we can rejoin at 12:55 sharp, since our next speaker from Biocon Bangalore will be joining. She has confirmed her joining through the link at 12:55. So Kapil, I think our links will be active. And
yes.
Zoom link will be on, so we all can join at not later than 12:55, so that we can start right on time. So we will break for lunch, and I request all of our participants to rejoin at 12:55 sharp. Thank you so much. Of biosimilars and its quality control. So let's hear from you, Dr. Jyoti.
Thank you. Thank you for the nice introduction. I'm going to put my video off and then start my presentation, just to avoid any network bandwidth issues. So in the beginning, I have a small correction to make, because I am now a part of Bicara Therapeutics, which is another subsidiary of Biocon. Okay. And we are focusing on novel biotherapeutic development, novel biologics. Okay. So I have joined this in Jan 2020. So I've kind of, yes. It's a new...
That's why you're so busy in evenings, so doing all those online meetings.
I'm sharing my screen, and I'm going to start sharing my video. I have a very ambitious slide deck, and I'm hoping to cover at least a few of the topics. But if we are short of time, because I'm not keeping a watch on the time, please let me know. Like, give me at least 10 minutes advance notice. That will be very useful. So as I mentioned, I am right now leading the product development for Bicara Therapeutics, where I have shifted to development of novel biologics. But in this slide deck, I'm going to cover a few topics on biosimilars. Very briefly talking about what are biologics, what are biosimilars, concept of biosimilarity. And then I have a couple of slides explaining why are we developing biosimilars, like what is all this craze about biosimilars. I'm also trying to give a bird's eye view of the complexity and the key aspects of development when we are developing a biosimilar. I have tried to give a snapshot of the biosimilar, and finally a couple of slides again talking about the very critical role of quality, compliance, and regulatory in the development of a biosimilar. That's why it's a very huge list of topics, but I'll try and see to cover, at least give you an overview of all of these today. A couple of slides in Biocon. We are a innovation-led global pharmaceutical company, and there's a lot of science-driven approach to the way we work. It's very heavily focused on innovation and science. We have made a global impact already with with a lot of biosimilars in the US and EU. Insulin being our main portfolio, along with biosimilar MAPs. So biosimilar insulins and biosimilar MAPs are our portfolio. We got our first trastuzumab antibody approval, monoclonal antibody approval in the US, and it was the first in world. And there have been multiple other approvals like pegfilgrastim, insulin glargine, insulin aspart, and all. We also are coming up with the next antibody, which is bevacizumab. The other key area for focus at Biocon is actually novel biologics development. We have quite a few molecules in the portfolio. This is not the complete list. And as I mentioned, there is incorporation of a new entity within Biocon. It's called Bicara Therapeutics. It's incorporated in Boston. It's a subsidiary of Biocon and focusing on the novel immuno-oncology molecules. So we are trying to get into the next generation requirement of therapy. So just a very couple of slides on what are biologics and what is the complexity that is associated. So as we all know, biologics are not chemically synthesized. They are manufactured in living cells. It could be mammalian, bacterial, or yeast. Most important to note is, like, when you're looking at a small molecule like an aspirin, it's like 180 Daltons. Whereas when you're looking at a monoclonal antibody, the size is significantly different. That becomes the first aspect of the complexity. And you have this intermediate biosimilars like an insulin, which is around 6,000 Dalton. A fact sheet again, a small molecule versus biologics. Pharmaceutical small molecules are chemically synthesized. They also have oral solids, liquids, and injections. Whereas if you look at a biologics, they're made by living cells, injected or infused most often, and it is dispensed only by doctors or hospitals. The size and the complexity is the major differentiator, because you can well define or characterize a small molecule, whereas the complexity and the heterogeneity, because of its manufacturing by a living cell, the difficulty to characterize, the challenge to characterize and consistently produce the same molecule, makes the manufacture of a biologic so difficult and challenging. So coming to biosimilarity, this is from the guidelines published by both FDA, EMA, WHO, and all that. A biological product that is highly similar to the reference product is called as a biosimilar. There is another definition which says that there are no clinically meaningful differences between the biological product and the reference product. That is, the product that you manufacture and the product that is already available should have no clinically meaningful differences. In a nutshell, the way biosimilarity is looked at depends on the totality of evidence. That is, there could be minor differences if it does not have any clinical impact in terms of safety, in terms of potency, in terms of immunogenicity, in terms of any risk to the patient of any type. There should be no risk that is linked to a difference. That means small differences are allowed as long as you are proving that it does not cause any clinical impact. So if you look at this slide, here it talks about the reference standard development. Sorry, I'm assuming that my voice I'm audible and there is clarity in the presentation.
Yeah, it's clear.
Yes, thank you. So this is how you would look at a reference product or a novel biologic development, where you establish clinical data over multiple trials, phase 1, phase 2, phase 3. You establish the safety, efficacy, immunogenicity in the clinic first, and then you would say that meanwhile you also establish the characteristics of the product. So here the clinical data is the bulk of development, whereas for a biologic, for a biosimilar, since this is already established, you're allowed to minimize the clinical development significantly. But on the other hand, you have to show that whatever the manufacturer, the reference product manufacturer have produced, you have similar, or the emphasis is actually more on the product development. You have to have physico-chemical and functional tools to showcase that the product that you have is identical or similar to the reference product already. You also need to have a very good understanding of the process and the sciences that are used to manufacture this product. So this is the key of a biosimilar development as compared to a novel molecule. So you can have an abbreviated clinical program, which again shows that the product that you are manufacturing, the biosimilar, is similar to the novel molecule already. So a robust product understanding, analytical controls to deliver a consistent product, and a comprehensive understanding of the process. It's called as process characterization. So this is the key for a biosimilar product development. Again, emphasizing a little more on what are the requirements. So for a biosimilar product, it must demonstrate biosimilarity with the reference product in multiple ways. First, as I mentioned, is the analytical studies. Show that the product is highly similar. There are no differences. Even if there are minor differences, they are not having any relevance to the clinical. Then you also do you also have the supporting animal studies and the clinical study. Here, the clinical study is more like again a biosimilar study where you do a head-on comparison, head-to-head comparison of your molecule with the reference molecule, and demonstrate that the safety, purity, potency of the proposed biosimilar product is similar to the reference product. And you assess the immunogenicity, the PK, the PD. So this is both of this, or all three of this together form the bulk of demonstrating biosimilarity. And quite a few factors can actually help when you're applying, because there is no one size fits all for developing a biosimilar. You can actually tailor the data requirement with like if you have a robust the analytical studies are robust. I mean, the strength of your comparative analytical study, whatever you're generating in the lab, and your process understanding is very robust. If you can show that there are very few analytical differences, then what happens is you're allowed to have smaller, I mean, the stringency of requirement for showing that there is no other clinical difference reduces. So the strength of your analytical data literally has an impact on the kind of stringency the agency will have on the analytical and the clinical and other aspects. So there is a allowance for tailoring the data. The next part, what I'm trying to cover is why is there so much emphasis? Why is there a craze about why is the industry moving towards biosimilar development? What is the reason? So all of us are aware of it, but just to summarize, biologics have actually revolutionized the treatment for many chronic illnesses, like for example, diabetes or autoimmune diseases like rheumatoid and arthritis, psoriatic arthritis, psoriasis, inflammatory bowel diseases, hemophilia. A lot of these areas of illnesses which are chronic, not just cancer, but all the chronic ailments have actually been, biologics are the key treatment options that are available today for all these chronic ailments. Of course, it is also highly, there's a lot of market space that is available today, and it's a growing part of the overall biologic. There's a lot of numbers that you can see. It is in billions of dollars, right? And everybody wants to be a part of it. The total number of, this is some data from some at least a year old. there are more than 200 manufacturers who are investing in this space and area now. That is, this is a highly evolving development area, manufacturing area, I would say. In Europe, for example, there are more than 77 biosimilar products that have been approved in all these product classes. It is not just in monoclonal antibodies, but if you look at it, there are a lot more product classes that are available. So it is both the requirement for new options to treat diseases, as well as the available of the next generation or the next way of treatment options. It has a huge benefit for patients, significantly huge benefit, because it increases competition. It increases competition for market space, and thereby it brings down, it has a potential to bring down cost. So this graph, for example, shows that by entry of some of the biosimilars into the market, actually there's a change in the price per day by introduction of market. So for example, 40% of reduction in the price by introducing of GCSF. This is again data from the Europe market. And also access, that is a lot more access is available to the biosimilar, to the overall drug market, because patients have options of choosing between the original product and the biosimilar product. And so there is a lot more product that is available in the market. And in certain countries in Europe, actually biosimilars have taken over completely in the market. India is yet to see the boom completely, but yes, we are also in a similar space, I would say. This is again a similar slide showing how the number of treatment days for one of the product like infliximab, which is for rheumatoid arthritis, has actually, so this is, the blue is the innovator or the reference product, and the yellow and the gray are the biosimilars. So as you can see, within I think 4 years of launching, almost 50% of the space has been taken over by biosimilars. But most importantly, can you see the uptake of product? That is, 40% of people are actually taking this drug because it has become more affordable. Again, another graph showing a similar approach where change in price and volume treatment days of the total market is shown here. And these are the different products. So the blue is the change in price, and this is the change in volume. That means so much more volume is being uptaken by patients. This is GCSF, this is human growth hormone, anti-TNF, which is for arthritis, and so on. So the quality of life is improving. I mean, you don't have to continue living with ailments and continue with the reduced quality of life. These drugs have brought in an option. If you are affected by a chronic ailment, the biosimilars are helping improve the quality. People don't have to struggle with living with a lower quality of life anymore. That is the main difference that the biosimilars have been able to achieve. Biocon has been majorly focusing on monoclonal antibodies other than insulins. And one of the reasons is also, if you can see, this is data, this is just a snapshot. There are 29 biosimilars. So US is still not in the mainstream. It's Europe, which is 77, where 77 biosimilars are already approved, whereas in US it is still 29 biosimilars, but more than 50% are actually monoclonal antibodies. So now coming to development of a biologic biosimilar, I had the opportunity and privilege to actually lead the trastuzumab biosimilar product for Biocon. I was leading the CMC, that is starting from process development until submission, meeting regulatory authorities, convincing them about the data that we have generated. All of that I had an opportunity. So that gave me a window to look at it from a very, very close angle. And what I'm sharing here today is the knowledge and information that I have gained during my process of developing trastuzumab biosimilar for Biocon. As we all know, we literally have to produce the same molecule as the reference product. So the biosimilar is actually essentially the same molecule as the reference product. it's complex. Why is it complex? Because you are trying to tell or make a living cell control what you want. I mean, you have to control the living cell and literally give it instructions. But what do you have in your control? You only have the nutrients, the growth environment, the bioreactor conditions. So a lot of uncontrolled aspect, but at the end of it, you need to have a similar product. Again, your product is also very complex. So a monoclonal antibody, for example, has 1,300 amino acids and associated manufacturing. So if you look at it similar like a cycle, if you are looking at an aspirin, whereas you literally have to produce an aeroplane when you are looking at a biologic. So that's the kind of difference that we are talking about. It's very complex, and to appreciate the complexity, I'm going to try and explain why is this so complex? What is the complexity due to? What is causing the complexity? So this is a cartoon of monoclonal antibody. These are the different domains. It has, as I mentioned, it has approximately 1,300 amino acids and many, many modifications. So what are the modifications? So one of the major modifications, for example, is the glycan that is associated. This is actually a very simple map. Now the industry has moved toward complex molecules, but this is a simple map with a glycan at the N glycosylation in the FC part. So the biological properties of the molecule, which could be because of the binding to the antigen caused by the FAB region. It has, many of the antibodies have also an FC function. That is, the FC part causes antibody-dependent cellular cytotoxicity. It is an effector-based function because of complement interaction, because of interaction to the FC gamma receptors, FC R1, R2, R3. So these are the biological properties of the molecule. You also have a lot of physical modifications. These are caused because of the cell environment, like it causes deamidation, oxidation, glycation, isomerization to many of these amino acids because they are exposed. The amino acids are exposed to the environment after they are, these are called as post-translational modifications. So all of this, so it could fragment, it could have many of these glycan variants are there, like it could be fucosylated, sialylated, and all of them can cause variations. So all of these are called as post-translational modification. And then you also have the structural variations. And then you have the molecules coming together to aggregate and all that. So what happens is you are looking at a highly heterogeneous mixture when you are looking at a reference product. So how do you develop a similar to such a heterogeneous mixture? That is what you do in a biosimilar development. First thing what you do is identify the critical quality attributes for the reference product. What are the key attributes which can impact its potency, safety, or immunogenicity? Characterize, understand the reference product as much as you can. Is the modification causing an impact to the potency? Is it an impurity, or would you categorize it under a related substance? So if it does not have an impact to the potency or safety or immunogenicity, so a lot of literature that is available that will help you determine this. So what happens is you can then categorize into, is it critical or is it a key, or you would say that it is a related substance. Then you also source multiple vials of the reference product because there is heterogeneity in the reference product itself. These reference products are manufactured at different sites. They use different scales of manufacturing. For example, a site A will have a 2,000 liter, where a site B could have a 10,000 liter. They source raw material from different regions. This causes a heterogeneity. Then sometimes you manufacture the DS, but you don't fill the DP. So the DS is aged, and that will cause another heterogeneity. So all of this also adds to variability other than the variability of the product itself. Then when we bring the product at our site, or we start analyzing that in R&D in our labs, we use a different method because these methods, what the innovator use, they won't publish it. So you have to literally develop your own methods and analyze the variability. So it's better to source as many vials throughout the development time as possible. It's not always possible, because if it is expensive, you can't buy a lot of products. And generally, these are very, very expensive, adding to the cost of development. As I mentioned, a good analytical tool is a key. I mean, if you want to understand the reference product, you have to have a good, robust analytical method early in development. So, an ELISA, for example, if you, this is from the public domain. As you can see, these are multiple lots of the same product, and look at the variability that they have. But some of the assays, say for example, ELISA brings its own inherent variability. So you wouldn't know whether it is because of the product or because of your analytical method variability. Another one, which is again talking about the variability of, it can be due to multiple reasons. So this again adds to the complexity of development. But getting a robust analytical method is very key, or one of the most important requirements for a biosimilar development, analytical and characterization. The other thing for a biosimilar versus a novel biologic is, so if you look at a novel biologic, what happens is you start with the discovery, you identify the molecule, and then you do a simple process development. But you can continue the process and product development as the product goes into the phase 1 and phase 3. This takes around 3 to 4 years. Clinical trial of phase 1 and phase 3 can take up to 3 years to 4 years at least, minimum of 3 years actually, both of this together. So what happens is, all this time is available for you to develop your process. CMC, which is basically your process and product development, quality, everything. For a biosimilar, before you start your phase 1 trial, you need to have a process in place. Your product that you're going to put into the clinical trial has to be actually biosimilar. Otherwise, your phase 1 clinical trial, if the product is different here versus what you use for the phase 3, and nowadays many people are not even, I mean, there is a requirement that you don't need actually a phase 1 clinical trial always. So if you do not have the final product, you may end up repeating some of the studies, and these are very expensive. So it becomes a mandatory requirement for the scientist in the lab to ensure that he or she has a final process and product before they start the clinical trial. So that kind of reduces the cost of development significantly. Snapshot of the process that is used for biosimilar development. As I mentioned, it is important to have a biosimilar clone. That is, when you're doing your clone development or your host development, you try and rank the clone based on the quality attributes. You would like to choose a clone which is giving product as close as possible to the reference product. So that's why analytical tools are very critical. Once you develop the analytical tools, once you understand the product quality of the reference product, then you simultaneously work on the clone development and try to get a biosimilar clone. This will also again save your time for development significantly. Then you have the cell culture and the purification development to meet the quality targets that you tailor the quality further. I'm going to give a very brief overview of the process development. So this is a cell culture just to show the kind of complexity that is associated with the upstream process development. You have to expand the host, you have to allow it to grow in multiple levels of inoculum generation at various stages. It depends on the kind of scale that you have at the production. But at every step, you need to test, is it growing fine, is it meeting the requirements or not. So each of these steps additionally have a in-process testing at every step, and all of this is first developed at R&D. This is the purification process. Again, multiple steps are generally there. In other than steps to meet the quality, meet the purity and quality are the main target. You also have steps to take care of the viruses. That is, these are redundant steps. In case there is a potential viral contamination, you need to have proven in that your process is capable of taking care of it. You actually have to do a study by spiking viruses, which is done in a virology lab, where you spike viruses in the process and show that your process can clear up to 10 to the power 6 of viruses. So you introduce steps. Say, for example, a low pH step is a viral inactivation step. You have a viral filtration step. These steps are only to make sure that if there is an accidental viral contamination anytime, because we are dealing with mammalian cells, these will be there to take care of your viral contamination. And then you have some of these steps also that can remove viruses. Again, here in process testing, so you have to remove the host cell protein, you have to remove the host cell DNA, you have to remove the media components, other than matching the quality of the product. And then the formulation is, again, if you have a product that is lyophilized, then you have more steps. If not, then you just if you want to make sure that it's a liquid product or a subcutaneous product, then you add your excipients and develop a formulation to maintain the stability. And so it is important to have the stability and shelf life similar to the reference product. Otherwise, it is not viable in the market. Point number one, it is a risk. Second is, it is going to be very difficult to get an approval for a product which does not have a similar stability or a lower shelf life than the reference product. The regulators will be
I think we have lost her voice.
She's back now.
Hello.
Yeah.
Sorry.
We lost your voice.
No, my internet conked off. Sorry about that. So did I cover this slide?
Yeah.
Okay.
You were
Thanks.
on this.
Yeah. So the next part of it is transfer of the technology to a large scale. Here you are dealing with pilot scale to large scale bioreactors. In the industry, there is up to 50,000 liter bioreactors that are available. But the general size is between, say, 2,000 to 10,000 liters mostly. And the pilot scale ranges from 50 to 200 liters. You also have very large columns. You start at like 300 millimeters 300 centimeter to 1 meter in diameter would be the size of the columns that you use at in the industrial scale. And then there is this large scale chromatography, skids, tubings. The major challenge that you face is time constraints. I mean, there's very limited time because the product is not stable. You have to ensure that everything is done as fast as possible. And then manufacturing in a controlled GMP environment. I'll just have a few pictures to show. So this is like, for example, a manufacturing suit. The particle count, I mean, you literally have class 100 to class 1,000, 10,000 manufacturing facility. It's a highly controlled environment. Hence, that again adds to, it's not a very easy manufacturing process. Complicated equipments, there would be many sterile lines and tubings. You also have to take, as I mentioned, there is testing involved, right? So you have to keep on taking samples intermittently. This is a picture from one of our manufacturing suit, which is for the mammalian culture. This is for the insulin. Compliance during manufacturing is a must. So other than the complication of keeping all of these active and going on sampling and everything, these people in the manufacturing facility have to understand the requirements of compliance and ensure that there is extensive documentation contemporaneously. This is where most of the audit observations come from, because our people feel that we can do it at a later point of time. So this the approach and attitude of compliance has to be a part of manufacturing, the manufacturing team. This is a purification suit. There are very large skids and tanks. I mean, it's actually quite strenuous to handle biosimilar or a biologic manufacturing. There are, this is a column, actually, this is a 1 meter column, and it is, again, I mean, it's huge, quite challenging and difficult. It is challenging, but it's also quite scientific, I would say. Engineering aspects are a must if you have to develop a manufacturing facility for a biologic. Other considerations are actually when you are submitting a dossier, you need to understand the process, design space, you need to validate the process, set specification, have control strategy. All of these are requirements from a regulatory perspective. So all of these aspects have to be taken care of for submitting a dossier. Some representative data from one of the biosimilars that we have submitted, so this is, the blue is the reference product, and the red and the green are the biosimilar product. Other physicochemical tools to reference. And I'm just going to show you some data from a biosimilar approval. So this was the trastuzumab that got approved. You have to show that it is identical. The amino acid sequence is identical. You have to cover every single peptide in multiple batches and show that the product is identical to the innovator. It is not easy to, the biosimilarity also comes from the potency and the other aspects, the bioassays, which are actually one of the toughest. You to establish the range for the innovator and then show that your product is within the range. So this is data which shows that with multiple lots of the US and EU product, the biosimilar product was within the range. Another bioassay, this is inhibition of cell proliferation. ADCC, that is antibody-dependent cellular cytotoxicity. These are the two mechanisms of action for trastuzumab. Additionally, because as I said, the FC receptor function can impact ADCC, can impact clearance, can impact PK, PD, and all that. These are also to be demonstrated in the lab to be similar to the biosimilar product. So this is a list of the assays. Around 35 assays were used. Some of them are orthogonal. Orthogonal, like if you see the protein transformation, we have used around 8 or 9 techniques for the higher secondary and higher order structure. The techniques establish the same attribute, but you need to use multiple techniques to show that the structure is similar. Or for example, here, if you can see multiple techniques to show that the HMWP content is similar, or glycoform, for example. So all of these techniques are for the same attribute. So it was 35 techniques establishing. It is literally like looking at the same picture using multiple different lenses. Finally, I'll just very quickly cover the other two important aspects. We spoke about development from an R&D perspective, but documentation and quality compliance is very, very, very critical. Actually, there is no shortcut to documentation. And this is actually the key, because your contemporaneous recording, GLP practices, including R&D, because scientists are not very good at this, especially R&D scientists. Manufacturing, you get trained and there is consistent training. So manufacturing and quality, it's easy to train scientists, but R&D scientists actually find it very difficult to keep writing everything in their lab notebook. So you have to train them again and again. And contemporaneous recording of data is very critical. So finally, what matters is a good and consistent quality system. In a nutshell, ALCOA, that is attributable, legible, contemporaneous, original, and accurate. This is what the quality system has to prove. This is what it should try and achieve. Some examples. So for example, what is this number? Is 29? Is it 27? Is it 24? Or is it actually 2%? So the recording of the data becomes so clear that when the agency comes for an audit, and if they get confused like this, then it can go into a very different tangent. So legibility, no overwriting. You just make sure that you're striking it once. You can't use pencils, you can't use markers, you have to write it with the blue ink. And if there is a correction, like for example, if there is a correction like this, a signature is mandatory. Otherwise, this is no longer valid. So all of these small things matter so much that finally this actually becomes a big thing. You may have a product that is very good, but if these small things are not taken care of, then you may end up not getting an approval. This is something that many of the students who come out from colleges and enter industry find it very difficult, because we in the colleges, we are not really trained for these. Then obviously the CGMP requirements, there's a lot of emphasis on everything in the quality. There are 6 systems of management, that mostly the Six Sigma system is what is used for quality management, facilities, equipments, material, production, packaging, labeling. All of these are covered by a very stringent quality system. And this is what we have learned in our audits. R&D is a very critical part of the story in an audit. And hence, sound technical knowledge. I mean, it's very important for the scientists to know what they are speaking. That makes a huge difference. It's literally a make and break kind of situation. Because if you're able to convince that your product is science driven, the process becomes easier. The final part is the regulatory part. It's a long and tedious process. I mean, you gain knowledge over a period of time. I mean, once you submit a couple of dossiers, which is what Biocon has gained the knowledge. I mean, and now I think we have 6 products that have been approved, which is very good for a company like ours. So what has happened, the scientists have understood the process, and we have a good wealth of knowledge with us. But it is a long and tedious process, summarizing the information, linking the correct information with each other. You have to review it multiple times and make sure that all the information right from the start of the development until the quality manufacturing release, everything is linked to each other. Then you can have multiple discussions with the agencies. FDA is very encouraging for you to have discussions with them. If you have any challenges or if you have any questions, they are available for advices. And this is also available with EMA, but EMA is more based on their guidelines. They have very well-defined guidelines. And then finally, the last one is the toughest, because once you submit, you have to be prepared for their questions, and answering those questions in a short period of time. In India, one of the biggest challenges that we all face is that, I mean, they work when we are sleeping, and that makes things very, very difficult, because this process, when after submission, where we are answering questions in a very short period of time, they give you very, very limited time to answer questions. So this process also is another hectic and strenuous process. So as I mentioned, the EU's biosimilar pathway is much better, because there's a lot of clear guidelines that are available in the EU. Whereas with the US, it is a highly interactive process. They encourage you to actually ask questions and keep in touch with them when you're developing the processes. So in summary, biologics and biosimilar development involves an extensive understanding of the molecular cell biology, process sciences, process engineering. I will emphasize on this again and again, because this is the key to scaling up and ensuring that what we develop in the lab gets scaled up and produces the same quality. analytical sciences, documentation, quality, regulatory. All of these spaces are equally critical and important in a biologics and biosimilar development. It is complicated and challenging, yes, but very highly gratifying is what our experience has been. Biocon has allowed, and we all, the team who has been a part of all these molecules that got approval, we have actually, there's a lot of satisfaction that we have today to be a part of a drug that is already approved and is going to be changing the way a therapy will be available in India, the cost and the volume that will be available and the access that will be available to the developing countries like India and others. So that's the last slide, and thank you. This is the Biocon Park facility in Bangalore. Kiran has developed an amazing facility, manufacturing, quality, R&D, everything. And this is the building that the R&D is located in, where I'm a part of, the Biocon Research Center. And so with that, I'm open to questions. Thank you very much.
I would request participants to ask questions if you have any to Dr. Jyoti.
Hi, Pradeep.
Hi, Dr. Pradeep. You are mute. Dr. Jyoti, I must say, I haven't heard such an excellent presentation on biosimilar. I have attended more than a couple of lectures on biosimilars. And this one is really satisfying because I think I am hearing from a person who has actually been such a part of actually right from the inception. What you are talking is, means I can generally feel myself that I'm working with that product, and that hard work and that tedious protocol which you have been showing, and the stringent critical part of quality control. I must say you must have gone, that journey itself is like, I can see from your presentation. It is really gratifying, and I must thank you for this presentation. And sincere gratification, I must say you must have gained. And I hope I have listened to your presentation, even what you gave at ICT. But I think that was a little different from this one. I've seen the YouTube video for that. It was a little bit different from this presentation. This was, I found it much more informative and more updated. A quick question from my side is that what are current pipeline products for MAPS? Which areas are being targeted currently? Is it cancer, immune therapy, or there are other areas also which people working with biosimilars are targeting?
So I would say it is still oncology and the Pradeep, please help me here. It is mainly oncology and the rheumatoid arthritis are the spaces. Diabetes anyway has been the focus for Biocon. So there's a lot of emphasis on insulin and insulin products and blocks also at Biocon. But the next wave of biosimilars is oncology and the chronic ailments.
Okay. I think Dr. Mayur has asked you a question. Yeah. Can you read those in the chat box?
Yes. So how about the reproducibility of biosimilars? What are the adverse effects seen in this? So reproducibility, I would say in terms of consistency of quality, this is something that we have to establish. If I can just project my slide again. I'm just going to show a slide which... Okay, so I hope this will answer your question. So as you can see in the slides, the red dots are the biosimilar product. And this is the reference product. So you have to actually manufacture multiple lots of your product and show that the quality remains the same. So using multiple techniques, you have to show that the quality, the range of the quality that is present in the innovator and the quality that your product has is similar. So as I also showed here, the number of tools that are used to achieve this. So we had actually around 9 to 10 batches when we were submitting our biosimilar application for trastuzumab. And that's also kind of requirement. I think you need to have more than 6 batches to show that you can reproducibly and consistently produce the same product quality. I think that would answer your first question. And the second question was, what are the adverse effects in biosimilars? So a part of the clinical trial package is showing that there are no adverse effects. The adverse effects are also supposed to be similar to the adverse effects seen in the innovator. So you actually are required to report all the adverse events that are happening in your molecule during the clinical trial and compare that with the innovator. So if there are any new side effects or if there are any new adverse effects seen, you have to prove that that is not because of the drug and it's not a part of the treatment at all. So there's a lot of stringency to the immunogenicity and the safety that is provided. That is also a part of the biosimilar development. Does that answer your question?
Yeah.
So the next question
is
yes, thank you. What does the future hold for the market biobetters and biosimilars? I would say it's biosimilars. The biobetters, at least myself, I have not really seen biobetter in the market. Pradeep, have you come across any biobetter?
Yeah, so biobetters are challenging because they will come under a different regulatory pathway. It will be considered as a new molecule and it will have a challenge of its regulatory process. So biosimilars is the quickest way to go for a molecule which is off patent. But if it's a biobetter, then you will have to go through the whole process of the new drug development. That's the regulatory challenge, right? So
thanks. What does the future hold for biosimilar? Okay, what about biosimilar and neurogenerative? I'm sorry, Priyanka. Actually, I don't think I have an answer to that question. The new neurodegenerative diseases, biosimilar landscape for neuro.
So if you're talking about Alzheimer's and this one, I think they are niche. There are not too many molecules in those.
Alzheimer's or Parkinson's, because that's also
Parkinson's, Parkinson's, yes,
Not too many. Not
too many molecules. Okay. They're all considered rare diseases. Not too many. It's an unfortunate part, but that is the reality.
Okay, thank you.
The next question is, what factors need to during peptide mapping study for biosimilar? So, can you please, Mithal?
Yeah, ma'am, sure.
Can you please repeat that question? I probably didn't understand.
During peptide mapping, we are taking it as one of the analytical perspective, which really compares the biosimilar product with the innovative product. So, which factors we are focusing during this peptide mapping?
So you look at modifications, any potential modifications, amino acid modifications. You look at post-translational modification. You also look at like glycan modifications. You have to look at each and every peptide actually and show that every peptide is similar to the reference product. So again, there was a slide in my presentation. I can very quickly again show that for your... How much time we have? 5 more minutes. Yes. So you can. So this is one thing that you have to show, that they are mirror image of each other. But additionally, each of these peptides are actually collected. Sorry, each of these peptide is collected. And you look at the amino acid sequence, you compare the innovator product with your product and show that the amino acid sequence in each of these peptides. So all of these multiple peptides in the heavy chain, in the light chain, right? This is only the heavy chain, right? What you can see is only the heavy chain. Each of these peptides, you have to establish that there is similarity. So one is the sequence coverage. Second is there are no modifications. There are no new modifications. The key is not having any new modifications.
Thanks a lot.
Is there...
Yeah, madam, shall I ask the question?
Yes, please.
Yeah, so say compared to the drugs, say biosimilars, I think the way they have a different approach strategy. Say whether we need to check for their acceptability, compatibility, maybe patient specific, or it is there are no such complications.
So when you design the clinical trial, Pradeep can also help me out here. I will, because he is more knowledgeable on this, I'm going to then pass the question to him. So Dr. Satish, when we design the clinical trial, we actually take care of some of these factors.
Yeah.
So the kind of population that is screened for the clinical trial, that is one consideration. But more importantly, since it's a head-on study of where you have blinded, I mean, it's a blinded study, right? It's a double-blinded study generally, where the patient doesn't know whether he's getting a biosimilar or a reference product. So that the design of the study where you have a head-on comparison between the biosimilar product and the reference product actually takes care of this risk of having an immunogenicity associated only with the biosimilar or a safety event associated only with the biosimilar. Because once you unblind and you collect all these events, you look at them in a comprehensive manner. You put the events under the that were due to the biosimilar product or due to the innovator product and compare them and see whether there are more number of adverse events or different types of adverse events or different types of immunogenicity that are linked to the biosimilar as compared to the innovator molecule. Also, the kind of PK assays, that is the PK and immunogenicity assays that are required for a biosimilar development are more stringent. That is the sensitivity required. So, for example, I will take the case of pegfilgrastim. There are two components to this molecule. One is the PEG, the other is the filgrastim. We were required to show that there is no immunogenicity because of the PEG. The immunogenicity assay that was developed had sensitivity to pick up any immunogenicity due to the PEG part of the molecule, not only the filgrastim. So you had to show that the two parts of the assays, both filgrastim and PEG, were taken care of separately.
Okay. Yeah.
Pradeep is,
yeah, I think you covered it. No
Yeah.
problem. That's...
Okay. Thank you.
I think we are out of time now, right?
I was just having one doubt here, Jyoti. That when we are saying 100% biosimilarity, 100% similarity, is there any window in your all complete 32 assays or characterization part, all studies I'm talking, is there any window where you have been able to not show data that it is 100%? What is that window? I want to know that. What is that window where is it like completely 100% similarity, or there is scope of some dissimilarity, which as a biologist or as a chemist or person who is doing this characterization part is having more understanding towards that so-called 100% or maybe 99.9%? What do you mean by 100% similarity?
So, Dr. Maushami, what I can do is just explain this question in a different way. What we do is we actually do a risk ranking. You are expected to do a risk ranking of all of these attributes. So, for example, let's look at aggregates or fragments or protein conformation, that's a secondary and high restriction, oxidation, charge variants, glycoform variants. We rank the risk that is associated with each of these variants and put them into different tiers. A tier 1, you need to have actually identical. You are not allowed to have any differences in a tier 1 requirement, which could be structure, sequence, it could be bioassays, for example, are in the tier 1 assays. There is no allowance for any difference. Then there is a second tier where there can be some minor differences, but you have to explain them. So how this taken care of is, so for example, you can see this range here, right? This range is from the standard deviation of this. That is, you look at the product variability in these innovator molecules, and you're allowed to have a 1 standard deviation, 2 standard deviation, 3 standard deviation, depending on the criticality of the attribute. So if the criticality is lower, then you can go, for example, for a 3 SD.
Okay.
But if it is more critical, then you need to have a 2 SD. So that is how they allow you to. So for example, fragments are not as critical as aggregates. Aggregates can cause immunogenicity, but fragments can impact potency. So you are allowed certain level, because there are other assays to test potency and you have a control on potency, you can have a slightly higher amount of fragments. Or for example, there are charge variants which are not having an impact on potency or safety. Those you can have some leeway, slight leverage is given.
That answers my question. Thank you so much. Yeah, I think I don't have, I can't see any further questions in the chat box. With such informative presentation, thank you, Dr. Jyoti, so much for being here in this workshop. And I was really looking forward for such presentation. And I think we have gained such a wonderful insight from you. And thank you so much once again from SPPSPTM, NMIMS. We also hope to see you sometime in our campus, whenever next time you are in Mumbai. So thank you so much.
Thank you for inviting me. It was a pleasure sharing the experience. I mean, one of the main things of gaining this experience and knowledge is if you get an opportunity to share it, it makes it more valuable.
Yeah, and a person who has actually worked with it is really makes it much experience, and we learn a lot. Yeah, it's your journey, so you have experienced really.
I really enjoyed it. Thank you.
Thank you so much. So with such a presentation, with such an informative presentation, I want to take the session ahead with Dr. Pradeep Nair, who is working as a Senior Director at Biocon Research Limited, Bangalore. He has immense experience in drug discovery group at Biocon's Research and Development, wherein his group has developed biological potency assays for drugs in the monoclonal biosimilar antibody program. In addition, their monoclonal antibody program targeted novel antigens. We are fortunate enough to have him today as a speaker, and today he is going to talk on cancer biomarkers and their potential clinical application. Dr. Nair's area of research includes cancer, immunology, and molecular biology. He has published many research papers in reputed international journals such as PLOS One, Cancer Medicine, Journal of Virology, etc. With such brief introduction, I would request Dr. Nair to speak to us and present his talk for the day.
Thank you, Dr. Maushimi. It's a pleasure being here. Thank you for inviting me here. And I think the screen share is on now. Let me just, yeah. Can you see my screen?
Yeah, it's visible.
Let's put it into, yeah. Okay. Okay, so thank you for this opportunity to speak here, and I will take you through the current developments in biomarkers in cancer. And it's a very interesting time in immuno-oncology and oncology space. And I think we are all witnessing some dramatic changes in this area of cancer biology. So I'll start with some few introductory. So I think so that we all get to, so it might get complicated a little more in the further, but you can interrupt me if you want at that point in time. But let me just point to options. Okay, so we have trillions of cells in our body, and all these cells will go through a cycle of life and death. So, so they all will die, and then none of the cells of the trillions which you are born with will be lasting the full life of your existence, right? So there will be damage when the cells begin to die, but there are mechanisms within the body to correct these damaged cells. That means phagocytes will come and eat this up, and so you don't see any further damage for these kind of cells, and you will have a healthy body. But by the sheer chance that sometimes, or unfortunately, maybe because of genetic reasons or spontaneous genetic mutations, because we as humans live a long life, there can be mutations which develop. And one of the very common mutations which happens is in the one of the which was discovered in the 1990s was P53, and almost 50 to 60% of all cancers have a mutation, and that mutation, and it was called the guardian of the genome. Probably you're all teaching it to students too. This molecule is still quite one of the key triggering events for cancer, in the sense that it is a cell cycle checkpoint molecule, which sort of decides the fate, whether it will go into a repair mechanism or it will go into the death pathway. Either if DNA is damaged, it will go into repair and go back to normal, or it will go into a apoptotic or a death pathway. So, but if this mutation takes place, as I mentioned, around 50% of all cancers have this mutation, or something similar in the same cell cycle checkpoint family, then instead of going to this decision tree, it progresses, and the cells carry this mutation forward and start to proliferate needlessly in certain tissues of the body, and that leads to other mutations to take place, because no longer is the ability of the cell to govern the mutation rate or the frequency of mutation, and the DNA polymerase makes an error 1 in 10 to the power, say 8 to 9, right? So, and the human genome is like 3 into 10 to the power 9. So you have chances of mutations taking place. And then you have additional mutations, and then you have a major mutations taking place that leads to uncontrolled growth and metastasis, which is cancer. So what are the different types of cancers? This is a typical slide which you see from the NIH or from this. And then immediately when you notice this, you will see some cancers which are common in India are missing. The most common cancer in India or in the subcontinent is head and neck cancer in men and women. That's because of the habit of chewing tobacco with betel nuts. And then in the women, it's the cervical cancer associated with human papillomaviruses. So these two cancers are sort of not present in the Western world. And so it's not mentioned in these kind of slides, but we need to keep that in mind also when we are looking at our population. The most common cancers in the West are in women is breast cancer. In men, it is lung, colorectal, and pancreatic. So you can almost all tissues of the body can get to become cancerous. So what happens when we're talking about biomarkers, we need to identify certain molecules which will So what exactly happens during this process? So this is a typical normal epithelial cancer, epithelial cell. That is here you see the dermis, and this is the epidermis, and this is the basal layer where the cells proliferate. And you can see a nice gradation. These will be the dead cells. This will be your outer epithelial layers, which are completely dead. So it will be a continuous turnover in this direction with blood vessels feeding in here. So this is the live epidermal. So then there is a transition which takes place that is called hyperplasia, where these cells start to divide, and you see this division layer are larger. And then you have dysplasia, where these cells look very different it's no longer similar to the normal morphology. It looks different, but still not cancerous. This sort of moves to a cancerous phase when it breaks the basal membrane and starts to migrate, and you don't see any differentiation at this level. So pathologists will score your tumor based on being benign or cancerous. This is what is generally the scoring pattern will be. And as I mentioned, in the Western world, the main causes of cancer is associated with tobacco smoking, that is 33%, excess weight and obesity because of associated diabetes and affecting cell division. Diet, as I mentioned, the lack of, there is a lot of studies which go about with high salt fast food diets causing cancerous, because it causes chronic inflammation, which can lead to cancer as I mentioned before, for head and neck cancer, you have betel nut and tobacco chewing. So these are the other viruses is also around 5%. There are some viruses which cause cancer, that is human papilloma viruses associated with cervical cancer as an example. And with the arrival of the vaccines in the Western world, this is also completely almost not to be seen there. In India, we still have a lot of cases of cervical cancer. So we have to work towards resolving that issue. These are very treatable issues, and these are needless populations being affected. So in the classification of the cancer types, you have carcinoma, sarcoma, myeloma, leukemia, and lymphoma. So probably all of you might have heard about it. The interesting thing is carcinoma is epithelial origin, and almost 80 to 90% of all cancers are of this origin. That means head and neck, breast, colorectal, lung. You can say most of the even brain gliomas are all part of the epithelial origin of cells. So sarcoma is in the muscle and connective tissues. Myeloma originates in plasma cells in the bone marrow. Leukemias are from the red blood cells producing bone marrow cells, so progenitor cells are proliferating rapidly. And lymphoma is from the glands or the nodes of the lymphatic system. So these are the major types of cancers that we deal with. So biomarkers, if you look at, there are around 13 to 14 biomarkers which are approved in the clinic so far. That is FDA-based approval, and they are used for multiple purposes. And I'll take a minute or two to walk you through this, because over many years, these biomarkers have been, refined, and probably you might have heard a few of them, like alpha-fetoprotein is with liver cancer. So this is very specific to liver cancer, and protein concentrations and the fucosylation content is measured. And this is used, the clinical application is for diagnosis, staging, detecting recurrence, and monitoring therapy. Then prostate-specific antigen, you can say, is very specific to prostate. And it's again for screening, discriminating cancer from benign disease. So you have another one, which is like OVA1, which is made for prediction, whether if this set of genes are proteins are expressed in ovarian cancer, so you can predict what could be the outcome of that patient's profile. So different biomarkers with different. Then there is this these three. This is a cancer antigens series are for monitoring therapy. Means in the blood sample of these patients, you can actually take a blood sample and see whether they are responding to the therapy or not, rather than having invasive techniques. So these are all approved So some of them are approved for more than one disease, like pancreatic and ovarian. Here you can see the CEA is for colon, gastric, pancreatic, lung, and breast. And again, monitoring therapy and detecting recurrence. This is HER2, which was what Jyoti was talking about. The Herceptin, the decision to give Herceptin to a particular patient, it decided by expression of HER2 protein. So if a woman is having breast cancer, it's tested for HER2 being positive at high levels, then the therapy of choice is Herceptin. So you can see the different ways. So it is for diagnosis, it is for predicting outcomes, it's for monitoring therapy, it is for therapy choice. So these are the different ways that a biomarker can help in making decisions affecting patient life. So basically, if you look at definitions wise, a diagnostic biomarker is something like I mentioned before, for a given cancer type, what to detect that. So somebody, if that particular protein is in abundance, it is used to say, okay, that particular patient has that particular cancer. For example, the presence of Bence-Jones proteins in urine remains one of the strongest diagnostic indicators for multiple myeloma. So this is highly specific to multiple myeloma. So then they have got prognostic biomarkers. This type of biomarkers is used once the disease status has been established. These biomarkers are expected to, give you what could be the outcome of this patient, because that person has this biomarker. So for example, again, in testicular teratoma, the HCG and alpha-fetoprotein can discriminate two groups with two different survival rates within the same testicular teratoma. So if you are having this, you have a different outcome compared to not having this. And then stratification predictive biomarkers are those which respond to therapy. Like I mentioned, the Herceptin is used for HER2 expressing tumors. So what happens exactly in this? So for a good biomarker to be qualified as a biomarker, it has to pass through the same statistical tests of specificity and sensitivity. So what you see here is a tumor marker value, some number, and the frequency of distribution on the y-axis. And typically, it will be a normal distribution when you look at population studies. So if a biomarker needs to be qualified as a biomarker, then it should be separate from this group, like what you see in blue. So this area is distinctly for patients who are positive. But then in this, you can see this section where you're putting a typical, if you say 5% cutoff, group C, which is the false positive. That means they were normal, but still diagnosed as a positive, and that can give rise to a lot of stress in a patient. Similarly, here you have this called a false negative, where you have this issue of the patient actually being cancerous, but not being detected because your cutoff is here. So an ideal biomarker should be spatially resolved. You should have this separation for both the sides. And this is also seen in the false positive rate, that is C, that is 1 minus specificity, and true positive. The higher this one is, the better the area under the curve, the better the biomarker. So this is typically what is used to qualify something as a biomarker. So you can't just say that I have some data and I will put it as a biomarker. It has to follow all these stringencies, and then probably FDA and the clinical the fraternity would accept this as a potential biomarker. So I will get into a little more of, these are the ones which were these molecules were the ones which are accepted in clinical, but the hunt for a good biomarker is still ongoing. And so I'm just taking an example from this paper which came this last year sometime, and this is talking about they're using a single cell pathology landscape for breast cancer. So in breast cancer, there are typically 3 kinds, that is hormone receptor positive or negative, then HER2 high or low. This is the only classification which is present for breast cancer. So that means there's a lot of cells which are uncharacterized, and that may lead to different, you probably are missing a good treatment outcome for these patients. So I will take you to this interesting paper. So, yeah, sorry. Okay. So what they've done in this is they have taken tissue. This is tissue microarray on a slide, and this is all got almost around 400 different tissue samples from different donors on one slide, and then they put a cocktail of antibodies. They have homed into 35 different antibodies, each of these antibodies being labeled with a metal ion. And this is taken through after imaging, they are taken through the instrument called the CyTOF. which is the mass plus flow cytometry. It is combined of both. And in the CyTOF, they actually quantify each of these protein expression. They put it into an algorithm which can actually resolve each antibody in each tissue. And then they can do a spatial community analysis. They do a tumor classification, and then they take it to survival analysis. And they have data from 20 years data. And that kind of clustering actually helped them to identify 27 different families of tumors from a population of around 250, 300 patients. So, and this is when they did the array, and you can see that in this set of groups, like this group is high in stromal compartment, whereas this group is high in the immune compartment. So you see that it is so heterogeneous, and so one treatment will give you the same outcome hypothesis does not hold, because you have so many subsets of cancer within this area of breast cancer itself. So, and this for people who are familiar, this is or not. This one is called the Tesny plot. This is like high data algorithm in a 2-dimensional array. When you put it, then you can see this clustering of clusters coming in, and this is how they identify these 27 different groups. So I just put this to make you appreciate that biomarkers is an ever advancing field. People still are finding out with using more refined tools about what is happening in the biomarker space. So even here you can see what is, and then they take these markers in this different groups. So you should see that the broad classification initially I told you was a HER positive, HER2 negative, HER positive, HER2 positive, HER negative, HER2 positive. HR means hormone receptor. Sorry, it's not HER. HR positive means hormone receptor positive, HER2 negative. So these are the 4 fundamental classifications which are present, which we are now categorizing. And you can see that this group, that is the dark blue, that is HER negative, HER2 positive, is the one which survives the longest. And this group. So HER2 positive is definitely a good this one, or HER hormone receptor positive. But this group is a poor category, that is HER negative and hormone receptor negative and HER2 negative. So this is an outcome, and this population is close to 50% of all breast cancers. What is the treatment option for these patients? This definitely, you have hormone therapy, you have Herceptin, you have other molecules, but for this, it's very difficult to treat them, and you can see that the survival is very poor, even in a Western country, and this is study done in Switzerland. So if you classify these, then you can see that inside these classifications also there are groups. I'm not going too much detail, but very interesting from this paper was one group which was actually classified as HER negative, HER2 negative, but they had apoptotic genes high, and their survival was quite high. That means with chemotherapy, you have and that is because they have apoptotic genes which are responding to chemotherapeutic drugs. So probably these patients, if they had been screened earlier, they could have, similar patients in different areas could have had better outcomes. So I'm just bringing in the point that we do not know too much of this biology of these cancers, and it's a work in progress. And similarly, you can see this is when you're talking about hazard ratio, that is anything less than 1 is good outcomes, anything worse than 1 is bad outcomes. And you can see very few criteria can give you good outcomes. Most of them give you bad outcomes in the current therapeutic regimen. So if you're grade 1, that means catch the tumor early. That means early stage, then you definitely are much better chances of survival. Your HER positive, hormone receptor positive and HER2 positive, you have high chance. Grade 2s are also better. But then this is the small subset of these categories which can respond good to the current therapeutic options. So so basically the biomarkers for targeted therapy today is for precision medicine, and you can appreciate that the first molecules which are targeting EGFR, that means if you are expressing EGFR, then you have treatment options with Erbitux and the small molecule inhibitors. And then if you have HER2 positive, then you have Perjeta, which can block the heterodimerization between HER1, HER2, HER3, HER2, HER4, HER2. And Herceptin, as I mentioned, which before for HER2 overexpressing, and this is Capzilla. This is ADC, which can bind, internalize, and kill the cell. And KRAS mutations, if you have, then probably you are not eligible for EGFR cetuximab therapy. So people now understand what is good. And the hot field is definitely immune checkpoint inhibitors. So if you are having PD-L1 expression on the tumor cells, you have treatment options with either an anti-PD-1 or an anti-PD-L1. So, and I will talk about it. So different cancers have different outcomes, and these are, and now companies, what they do is, before they go into the commercial launch of a molecule, they come with companion diagnostics, which is approved by FDA, and this is focused on biomarkers. And you see the largest number of companion diagnostics is with the HER2, because that's it was launched in 1998, Herceptin, and it's got the largest millions of people saved. So it's a wonder drug, actually, you can say, because, and this is all associated with companion diagnostics. Other indications have one each or two based on the different kinds of cancer types. I'll shift gears and I will talk about immune checkpoint therapy. This buzz in the field started with a landmark paper which got published in 2011 in New England Journal of Medicine, which talked about in metastatic melanoma, which had a 5-year survival rate of only around 10%, that people had actually lived more than 70%, more than 30% of that population survived for over 5 years. So that was a big change, because metastatic melanoma is a very complicated disease, not seen very commonly in India because we have melanin pigment which protects us from UV radiation, but it's very common in the Western world. So James Ellison, working independently in 1990s, pursued the path of CTLA-4 biology, and CTLA-4 biology comes from this, where you have a T cell and a antigen presenting cell. When they interact, the T cell will present a TCR and MHC. This is the first trigger, but that's not enough to activate the T cell. So the second signal is CD28 and B7. So once this T cell gets activated, typically for when it sees a microorganism or against a virus or some infection, then it has to be, after the infection is over, it needs to be suppressed. And so these T cells release a molecule called CTLA-4, which CTLA-4 can bind to the same B7 molecules and suppress. So it will compete with CD28, and CTLA-4 will outcompete CD28 binding to B7 and suppress the T cell. So it's a suppressor. So now if you come with an antibody, and that's James Allison's very interesting point, was if you come with an anti-CTLA-4 inhibitor, you block this interaction and you can keep this T cells active. So that's the whole idea. So same thing was done by Sumo Hojo from Japan, where he discovers this new molecule called PD-1, which is again expressed on activated T cells. This PD-1, PD-L1 interaction, PD-L1 is typically overexpressed on tumor cells. If you block this, then the inhibitory signals to this TCR is released, and these T cells get active and can kill these tumor cells. So that's the hypothesis. The Nobel Prize was given in 2018 to both of them for discovering this new way of taking the brakes off the immune cells. This is data from the same checkpoint study, which was because nivolumab was the anti-PD-1, and ipilimumab is the anti-CTLA-4. So nivolumab came after ipilimumab because both are from BMS, Bristol Myers Squibb. Ipilimumab targets CTLA-4, nivo targets PD-1. So this is data from the anti-CTLA-4. This, for those who don't know the survival curves, this is on the x-axis is time. It can be months, years, etc. So here it is months. So you're talking about almost more than 4 years of survival. And y-axis is overall survival, and it's as an event is a death event. So if there is a fall, then it'll be relative to the 100% survival at baseline. It'll taper off. And this is what you got. So typically, metastatic melanoma used to survive like this, and only around 5% used to live for this time. So you see this gap is what people are talking about. This is the CTLA-4, and this starts from around 2 years of time. That means metastatic melanoma otherwise would have gone like this without the immune checkpoint inhibitor. So this is dramatic, and so many millions of people have survived because of this. This trial actually talked about combination of nivolumab plus ipilimumab is here, and nivolumab, that's anti-PD-1, gives you even better than what is given with CTLA-4 with less toxicity. And that's the interest. So nowadays, people don't use ipilimumab at all because the PD-1, PD-L1 axis is much more safe and more tolerable. But the point is, how many people get advantage? Because you see here, there is a what? So we're talking about 40%. 60% don't get any benefit, correct? Only 40% get a good chance of survival. So how do you get to ensure that the patient population is correct and you are benefiting or not? And so that's where these markers have come in. And these markers are called, the main marker is actually PD-L1. And so the PD-L1 expression can have different patterns, that is expressed very highly, moderately, or only expressed in immune cells, not in the tumor region at all. So criteria for this are like PD-L1 is TC3, TC2, TC1, and TC0, where TC3 is like greater than 50% of the cells expressing. And then you have these cutoffs. And then for immune checkpoint, immune score, there you get greater than 10% and all this. And then you have a combined score with the pathologist will score and tell you, okay, are you eligible for treatment with nivolumab or not? So, so what's happening is this is a survival curve. Why the emphasis on this? Because if you if a patient is positive for PD-L1, this is the kind of survival. That means 50% is around 9 months survival, whereas 50% is only 6 months survival for a non-stratified, non-qualified, characterized group. So this is what you see with, again, with this is with naive lung cancer. Very true with metastatic melanoma too. And this is not only in progression-free survival, but also in overall survival, you see this difference between patients who are selected and patients who are not selected, the overall population. This difference that you see. So which is the, so if you're 80%, if somebody is 80% and more of PD-L1, you will benefit from that. The question mark comes, what is my negative cutoff? Is it so? Some people say 1% and above, and some people say 25% and above. So there is no clear definition of a good cutoff of how much immune histochemistry positivity can translate into a good biomarker and clinical efficacy. And this is exactly seen here. That is, this is the dark line is without stratification, and this is with combined is what you get with stratification of patients. So 26% patients respond with atezolizumab, which is an anti-PD-L1, whereas 40% respond when you select for PD-L1. But then this is true for many other 15, 26, 13, 23, 20, 31. So 20 without PD-L1 selection means all the population, and 31% outcome when you select patients based on PD-L1. But then the problem is, what about the rest? You're talking about 30-40%, but there is 60% who are still not getting any treatment because they don't fit into any of these criteria. So there must be something beyond PD-L1, and that so Beth Vergilstein and others actually talked about this in the mid 2000s, and they sort of predicted that one of the things could be if there is more genetic tumor mutational burden, then the chances of neoantigen presentation would be higher, and therefore the immune system will be more activated, and there can be more effect of immune checkpoint therapy. So to test the hypothesis, they actually screened for different cancer types based on mutational burden. And so to use that, they use the exome, the tumor mutational burden. So they take the normal tissue from the same patient and the tumor tissue, and then they do the full sequencing. And then what the amount of difference that you see is the mutations which have come in the tumor region. So that when they compared, and then they also compared it with kits, which are key genes, which can, like I mentioned, P53 and similar, there's a set of genes which you don't have to look for the whole screen, because this would be a very, very expensive proposition, right? Even in the US, it would take around $3,000 to $4,000 to just do this test. But the smaller kits could be more useful, and there's a good correlation. And this is also seen in other tests, whole exomes versus test mutations per megabase, mutations per megabase. So you get a good correlation. And this is actually seen in this nice paper which came in 2017, that if you put on this x-axis number of coding somatic mutations per megabase versus objective response rate, these tumors are highly responsive to immune checkpoint therapy. They definitely have more mutational burden. That means they are significantly different from the normal phenotype of these within these patients, whereas these are very poor outcomes, and you can see pancreatic, mesothelioma, sarcomas are all poor outcome tumors. So this is the fact that some of these are driven by some particular mutation, driver mutations, and this are more, more established tumors, probably established over longer time, have more chances of multiple mutations, and therefore more neoantigen presentation, more immune response, and better response to immune checkpoint therapy. So this again was confirmed in other studies for biomarkers. So if you stratify the patients based on T-cell inflamed gene expression profile on this axis and tumor mutational burden, you actually see it as 4 different populations. One, which is a very cold tumor. That is, there is hardly any tumor mutational burden or any inflammatory molecule within this. This have the extremely poor response, and that is group A. It's saying overall response rate is less than 10%. This has high tumor mutational burden, but not too many immune cells infiltrating on this. And this is just above this, so something between 11 to 30%. This group has the gene expression, more immune cells, but less of tumor mutational burden. And that comes under C, which is 12 to 35%. The best responders are this group, where you have immune cells plus tumor mutational burden. And this was very interesting that these kind of patients can benefit from these expensive drugs rather than treating everybody in this population with the same drug. The other interesting thing is nowadays there are a lot of studies which show that what you eat and what you, this one has a big influence on how you respond to immune checkpoint therapy. So there are some friendly bacteria which is there in your gut, which can influence the immune checkpoint response. better than certain other bacteria, which probably will have a better, worse outcome. And this has now been carried forward. The studies which came out 2-3 years back has now been carried forward to even look at autoimmune diseases, how gut bacteria affect diseases in the brain and also in autoimmune diseases. So, so today what is happening in the field is like, these are the number of studies which are taking place. This is actually a little dated because it's over a year ago. And more than 4,000 trials going on with different combinations of immune checkpoint inhibitors. And each of these patients anyway is between $50,000 to $60,000 being spent per patient. So you can see the amount of cost which is being bumped into understanding and trying to find a cure for different kinds of cancer. This is different kinds of cancer, any cancer actually. So, and where this space is. So I will take this slide just to say where we are today. And the reality is that with the advancement from the 1990s of precision medicine, like molecules like Herceptin, before that even your Gleevec for ALL, and the hormone receptor therapies, tamoxifen for breast cancer, Herceptin for breast cancer, and precision medicine with small molecule inhibitors and molecular antibodies. And now the advent of immune checkpoint inhibitors, cancer has really become more chances of survival. You can see this is this was the original in the 1970s and 80s, especially for, if this is typical from metastatic melanoma, that hardly anybody survives over 5 years. With conventional therapies, with chemotherapy, this was the expectancy, but overall survival was still very poor. But with immune checkpoint inhibitors, as I mentioned, around 30% of the population live very long and almost cured, which is different. The whole field is moving towards combination therapy based on biomarker selection, and based on response therapies. You can see monotherapy is like 10 to 30%, combination is 40 to 60%, and the adverse events is what people are trying to address, because as you build in more combinations, you get more safety implications, but clinicians are getting a handle onto that. So this is my last slide. I will take questions. Thank you for this opportunity, and I hope this was useful. Thank you. Hello.
Yeah, we'll wait for a couple of questions.
Yes, please.
Meanwhile, I have a question, Dr. Pradeep.
Yes, please.
With this emergence of genomic profiling era, we are currently in this genomic profiling age. So I was just wondering how this genomic profiling thing is working with cancer biomarkers in current scenario in Indian clinical management of cancer patients. Is it contributing to any extent, particularly in an Indian context?
Yeah, that's what I said, that in the Western world, the exome sequencing and these kind of tumor mutational burden kits are available or are beginning to come, but still this is an affordability issue in India. And I am not very much aware about what is in the Indian scenario. Probably somebody from ACTREC or somebody can comment. But as far as my knowledge, it's not routinely used in clinical medicine in India.
Yeah. Questions are invited from the participants. Do we have any?
Yeah, so Mittal here. I
Yes,
just
please.
posted question in chat box. So can you please emphasize more on that metabolite study can help to identify biomarkers?
Sorry, could you please repeat that question? I sort of missed that.
She has put it in the chat box.
So my question is,
Okay,
in
chat box.
detail
Let me open it.
metabolite study can help to identify any biomarkers?
Okay. I am not aware of the ones
So
which
you're
are.
not audible.
Yeah, it's audible. It's audible. So
yeah.
So these are the kind of metabolite, this one, which are currently approved in the. Can you see my screen?
Yes.
Yeah. So these kind of molecules or proteins which are coming are in the accepted this one, but beyond that, I am not very sure. So some of them you have probably heard of, like prostate-specific antigen or CA115, which is a breakdown product of carbohydrate antigen. This one. So these kind of molecules are definitely in clinical practice, because the bar for a molecule has to be this, and it has to be. It cannot be that the normal group also covers a large range. So you need to have that, and the stringency of that cutoff is very, very important for being as a biomarker.
Thank you, sir.
Maybe I'll take. Okay.
There is a query from a participant.
Yes, please.
Are you willing to share your slide, your presentation? Someone is asking to me.
Okay. I'm not sure about that. Let me just think about it.
Yeah, you can just let us know.
Yes, please.
Okay, so if we don't have any further questions, Dr. Pradeep, I'm really grateful you spared your valuable time and spend more than an hour with us. And I'm sure this area of cancer biomarker is continuously evolving, and there are a lot of things which are still going to come. And it might enable us for early diagnosis and proper treatment of several malignant conditions. We all have cancer patients around us. Every alternate family, every other family, we are facing such situations in life. So I'm sure that in future we might be coming up with early diagnosis that will really help each one of us personally and in community at community level. Thank you so much for an excellent presentation. See you sometime in future once again.
Thank you.
Thank you.
Thank you.
Bye. Dr. Mayur, are you around, sir?
Yeah, I'm around.
Sir, we will have to wait for Dr. Peters, I guess.
Yeah, 3 p.m. So I think it will be 10:30 there.
So maybe next 10 minutes we should expect him.
Yeah, I have just sent a WhatsApp message. Let's see.
Okay. We'll wait for our next speaker. We can be in mute and wait for the next speaker. In next 10 minutes, he would probably join us.
तो तब खर्चे कम थे।
Hi, Fritz. Good to see you again.
जेजे पीटर का शुरू हो गया।
Are we audible, Fritz?
He has to unmute.
Yes, sir.
Yeah, you can hear us, Fritz.
I can
Good
hear you.
morning, because it's 10:30 in Amsterdam.
Yes, almost, yes. You
Good
can hear.
morning. Good morning, Fritz. Good morning, Dr. Peters.
You can hear me because I hear a noise in the background.
You are audible to us?
Can you hear us, Fritz?
I hear you, but I have I hear a noise and I hear an echo.
Echo, okay. Now, is it clear? I think, can you mute Moshmi and Moshmi? Yeah.
I can mute you.
You mute yourself and
only you.
So now, is it clear, Fritz?
No, there's still.
But you are clearly audible to us.
Oh yes, but okay. But as I mentioned, I hear now it seems to be gone. Yes.
No, it's on. Yeah.
You are waiting for me?
Yes.
Oh, I was just checking whether my slides on this computer were okay or not. So that's why I was not yet logged in. But so I want to see whether everything, because I made my slides on a different computer and I always have to check whether they will do fine in another one. But they were. So from that point of view, this is fine with me.
Okay, we'll start. We'll start for the next session. For the next session, we have a renowned professor in the field of cancer research with decades of experience and knowledge, Dr. G.J. Peters. He's a professor of pharmacology at the Amsterdam University Medical Center, Amsterdam, and professor of biochemistry, Medical University of Gdańsk, Poland. He has worked extensively in cancer and related therapeutics. He has done extensive research in cancer resistance and diverse areas of steroidal compounds and their role in cancer treatment, kinase inhibition and cancer treatments, and many more. He has more than 1,000 research articles to his credit and with more than 41,000 citations. His research work is of paramount importance in the field of cancer research, and he has published in many internationally reputed journals such as Autophagy, Proceedings of the National Academy of Sciences, Bloods, and many more. I welcome Professor G.J. Peters for his talk on co-cultures of three-dimensional spheroids as a model for drug resistance. Dr. Peters, we are eager to listen to your exciting talk, and we are going to hear you for next 45 minutes or so, and followed by 15 minutes of interaction.
Yeah, did you share me?
Yeah, you can share your screen with us.
You can see my slides?
Yeah, it's visible.
Okay, then. I wanted to increase the size of my mouse, but apparently that doesn't work yet. Anyway.
I think you are on the last slide, Fritz. I think you have to go back.
Yeah, that's why I was. I said I was checking my slides. No, why? My it's. My mouse doesn't react to what it should do. No.
Sir, can you directly press F5?
No, it's
now it's okay. Yeah, now it's okay.
Yeah. I don't know what went wrong compared to what I usually do, but possibly anyway. On my screen, the taskbar is covering part of my slides. Is that happening with you as well or not?
No, your first slide is clearly visible to us now.
Oh, that is okay, because sometimes this is happening with when you present and sometimes not. Anyway, what I want to do today is to discuss some of our work on pancreatic cancer and the use of three-dimensional model systems for this purpose. As mentioned, I am professor at the university, the VU University in Amsterdam, now called Amsterdam University Medical Centers, and I also have a position at the Medical University in Gdansk. Most of the work which I'm going to present today is done in Amsterdam, and you will give you in my next slide an outline of what we're going to talk about. I will give an introduction on pancreatic ductal adenocarcinoma, on three-dimensional culture systems, how pancreatic cancer cells are growing, what is the role of supporting cells called pancreatic stellate cells, and discuss the efficacy of a drug extensively used for treatment of pancreatic cancer and some novel drugs called c-Met inhibitors. I will discuss the morphology of the cells, the sensitivity and combinations, and I will end with some conclusions. Pancreatic ductal adenocarcinoma is a disease which is has very poor prognosis. The 5-year survival of pancreatic cancer is less than 8%, and this has already been the case for the last, actually forever. We have been treating pancreatic cancer cells. When I started to work on this, it was just 5%. There are several reasons for this. The detection of pancreatic cancer is very difficult, both from a medical point of view and from a genetic point of view. We don't have biomarkers. The patients are being diagnosed when the tumor is already spread and metastasized, or it has already an advanced stage at the local site. Another important point, which I'm going to discuss today as well, is resistance to actually all the type of drugs which we have. Sometimes the patients are sensitive in the beginning, but usually they become resistant as well. The current therapy of pancreatic cancer, or as I will call it, pancreatic ductal adenocarcinoma, is surgical resection, but this is usually feasible in only a minority of patients, 20-30%, and this is a very complicated surgical procedure. Usually adjuvant chemotherapy is given, which is a complicated combination called FOLFIRINOX, which is a combination of 5-FU, leucovorin, irinotecan, and oxaliplatin. This is a combination treatment which is quite, as I've mentioned, complicated, but is also stressful for the patient, causing quite a lot of toxicity, and a large part of the patients will not tolerate this type of treatment. Gemcitabine, either as a monotherapy or in combination with nab-paclitaxel, is also widely used as adjuvant chemotherapy. This type of therapy is better tolerated by most patients, and when the combination with nab-paclitaxel is not acceptable, it's only gemcitabine which is given. I will discuss the mechanism of action of gemcitabine in some of the later slides. Drug resistance is one of my major interests in cancer research, and the drug resistance in pancreatic ductal adenocarcinoma are being studied at different levels in our department. We are looking at DNA sequencing, RNA sequencing, microRNAs, proteomics, immune cells, and the microenvironment, and these are all marked in red. We ask ourselves, do we have the right models for this? Do we need human xenografts, or can we use orthotopic models? And with orthotopic models, we mean models which usually they are mouse models in which the tumor is implanted at the site of the pancreas. And so they are using the pancreatic environment. There are also syngeneic models called GEM, not to mix up with gemcitabine, and GEM stands for genetically modified mouse models. And we have three-dimensional models, and I'm going to discuss this in the next slides. So we want to understand how the microenvironment of pancreatic cancer influences the growth of pancreatic cancer. And we ask ourselves also whether we are using the right models. Usually for drug screening, we use two-dimensional models. And in these two-dimensional models, you can study the role of conditioned medium. And what I mean with conditioned medium is medium of these supporting cells. I'm also going to discuss three-dimensional spheroid. I'm sorry for this typing error I see. And we have been using cultures of pancreatic cancer cells and supporting cells. Why are we using three-dimensional models? As I mentioned, the standard drug screening is usually done in two-dimensional models. However, the three-dimensional models is more closely to a tumor. And here you see a picture in which the tumor is schematically given. So here we have the tumor, and in the middle of the tumor, there is usually a necrotic region, and at the border of the necrotic region, there are quiescent cells. Quiescent cells are cells which are not dead, but they are also not growing. The growing cells, the proliferating cells, are at the border of the tumor, and these cells are also bordering the vascularization. And here we see a vessel, and you can imagine that the nutrients can diffuse into the tumor, and only to a limited extent, and only the cells which are very close to the vessel will get sufficient nutrients and oxygen to survive. So the vascularization of a tumor is a limiting factor. What is not very clearly shown in this picture, but I will go back to this later, are the immune cells, which are penetrating from the vessel into the tumor, and supporting cells. Immune cells, for instance, we have tumor-associated macrophages, which are also located here in this area. What do tumor cells and these supporting cells do? They secrete, for instance, growth factors so that the vascular endothelial cells can sprout into the tumor and form new vessels. I will give a little bit of a short historical overview. We have a number of three-dimensional model systems. There's some so-called hollow fiber assay, which means that we use a hollow fiber in which the cells are growing and they can reach a three-dimensional system. In the 70s, tumor colony formation assay was very popular, and this was done by taking tumors from patients, making a single cell population of this and grow them on agar plates, and then they would form colonies. Very close to that are aggregates, and sometimes colonies are called cannot be recognized in a different way, and some aggregates are sometimes called colonies, while they are not really colonies. We have developed a model system which in V-bottom systems, which I will explain in a minute. Very popular at this moment are organoids and spheroids. Organoids are tumor, not only tumor, but also normal cells which are coming from a normal tissue and which resemble the normal tissue in their three-dimensional structures. Spheroids can do the same. Sometimes you could actually call spheroids organoids or organoids spheroids. We have been working with spheroids more extensively because they have a three-dimensional structure, and our spheroids, at least that's the one which we have used for pancreatic cancer, resemble the tumor quite in a lot of different aspects. The organoids which I've seen for pancreatic cancer, for instance, are more fibrotic tissue, so I'm not yet convinced that the organoids for pancreatic cancer are really the right model systems yet, but they are improving, and maybe in a short period from now they will have better model systems. I give you some example. Here we have a tumor cell aggregate, and what you see here is that this is the bottom of a culture plate, and we have a number of different cell lines. You will see at some point that the cells are aggregating, and they will form a three-dimensional structure, as you can see. What you can see here is also a little bit of a necrotic area. I just want to mention, if you have any questions, you can do this, or you can put some remarks on the using the Zoom option. You can also form aggregates, but in the three-dimensional model systems, and that will be shown in the next slide. And you remember here the previous slide, which I showed as an example of how a three-dimensional tumor is looking, although of course you see the two dimensions here. But when you make a cross-section of such a tumor, like this, a V cross-section, then you get a picture like this. And in vitro, you will have here a necrotic region, you have a quiescent region, you have proliferating cells, and instead of the vessel which is around the tumor, we have the medium surrounding the cells. When you have V bottom, it's the medium which is only here, and so these are the proliferating cells. How are we doing this? This is a normal cell culture plate, and those of you who have been doing cell culture may recognize this. For those who are not familiar with that, these are 96 wells, 12 wells here and 8 wells here. And what we are usually doing is culture cells in these wells. They have a volume of about 200 microliter. And what you see here is the medium in the wells. What we usually do is don't use the outer wells because they are more likely to evaporate. So that means that in the outer wells the volume is decreasing in time. You have different bottoms in these wells. We have a flat layer bottom, which is simply looking like this. We have a round bottom, which is simply looking like this with a round bottom. Usually these flat bottoms are used for monolayer cells, and the round bottom wells are used for suspension cells. However, there are also V-bottom cells, and they have a point-like structure. And what is happening is here, you see here the flat bottom cells, here have the round bottom, and here we have the V-bottom. And the V-bottom cells, wells, can be used to form multilayers. So what we do is put cells into the V-bottom. They are going to proliferate at some point, and you get a structure like this. And you can imagine that here in the point of this V, the access to nutrients and oxygen is less. And that's indeed being shown in this picture. Here we see an example of a colon cancer cell line, SW620. And here we see the cells in the bottom. Here we have the cells which are facing the surface. And what you can here actually see is a necrotic area. And these are the cells which are proliferating. The advantage of using such a system here, this is a 96 cell well system, is that you can do when you don't use the outer wells, so the 12 wells here and the 8 wells here, you can use about 80 wells to do culturing simultaneously. And what we usually do is have 3 wells as a triplicate for drug sensitivity and 3 wells as a control. And when you want to follow the growth of these cells, you can color them with a stain. We usually use a SRB for this. We validated that the SRB is indeed penetrating into the bottom of this stain, and SRB is a protein staining. So this makes it easier to do relatively large scale screening. I'm not going to in all the details, but these 3-dimensional model systems have molecular properties which are resembling a tumor. They have a decreased proliferation. They have hypoxic areas. They have necrotic areas. They have a decreased S-phase accumulation, so the cells are, which fits with the decreased proliferation. The proliferating enzymes have a decreased expression, which we investigated both on the messenger RNA level and on the protein level. Since cells are dying from necrosis, they are also more, some of the proteins are increased. And most of the drugs which are targeted against proliferating enzymes have a decreased sensitivity. And we also have investigated the accumulation of the drug and the penetration of the drug. And I will show you some examples of some of these properties. This is a characteristic growth curve for these cells. We have here an A2780, which is an ovarian cancer cell lines, and the SW620, HD29, and WIDR are colon cancer cell lines. Here in the left picture, you will see a normal growth inhibition curve. What you see on the y-axis is the number of the relative number of cells, and on the x-axis is the concentration of the drug in, or in this case, gemcitabine called DFDC in molar. So this here is 1 micromolar, and this here is 1 nanomolar. And you can see that the sensitivity of these cells is in the order of magnitude of about 10 to about the most resistant cells, 100 nanomolar. However, when we look at the right side of this picture, we see the sensitivity of the 3-dimensional model system. And here we see the 82070 cell lines, which we included since it's known to be sensitive in a lot of different model systems. And again, it was sensitive via the gemcitabine as well. But the 3 colon cancer cell lines are much less sensitive. And what you see here actually is that they don't reach a 50% growth inhibition. And even at a very high concentration of 1 millimolar. So we use a different way of expression of the sensitivity of this, which we call not the IC50, but the EC50. And we could calculate these values for a number of different drugs. And when we don't reach the 50, then we call them higher than. For the SW620, some of the drugs which we tested are summarized. Here we see the gemcitabine again, and I said we called this the EC50. This is 1 micromolar. Here we have for gemcitabine in this cell line IC50 about 100 to 200 nanomolar. But what you can see here for gemcitabine, the concentration to reach 50% inhibition is around 1 millimolar. So this is really sky high. For 5-FU, this is even much higher. For another drug called doxorubicin, it's less, and the difference is also less. And very interesting, there's a drug called EO9, which is a drug which is actually dependent on hypoxia. This drug is activated under hypoxic conditions, and what you can see is there's not much difference. This drug has also a low difference between HPC, which is also a drug which is less dependent on proliferation. Here we have cisplatin, and also the difference for cisplatin, because it's less cell cycle dependent, is less than for gemcitabine. Rhizoxin is a natural product which we was testing at that time, and is very active under normal conditions, but quite inactive under in this multi-layer system. Here we have some topoisomerase inhibitors, which are also dependent on proliferation. We see a large difference. For these 2 drugs, the topoisomerase inhibitors and the gemcitabine, I will show a little bit more details. Here, this is gemcitabine, and I go a little bit into the mechanism of action of this drug because it's coming back later. Gemcitabine is a nucleoside analog. Here we have the base part of the nucleoside. Here we have the sugar part. And gemcitabine is cytidine analog, is modified here at the 2' position. The 2' hydroxy group has been replaced by 2 fluorine atoms. And this completely changes the properties of this compound. It behaves as a deoxynucleoside, so that means that it can be incorporated into the DNA. But since this hydroxy group has been replaced by 2 fluorine atoms, it also behaves like a ribonucleoside. And that's shown in the next slide. Gemcitabine is a compound which can pass the membrane by diffusion, but an efficient uptake is being mediated by transporter called equilibrative nucleoside transporter or concentrative nucleoside transporter. So this concentrated nucleoside transporter can transport the drug into the cells against the concentration gradient. Gemcitabine is activated in the cell by several kinases, nucleoside kinases, and then the active metabolite is being formed, which is called the triphosphate, or I will also call it DFDCTP, difluorodeoxy-CTP. This triphosphate can be incorporated into DNA, and it will compete with deoxy-CTP for incorporation into DNA. At some point, this incorporation is so extensive that the DNA will not proliferate, will not duplicate anymore, and the cells will die. Interestingly, gemcitabine has some additional actions, and one of them is that the diphosphate will inhibit ribonucleotide reductase, and ribonucleotide reductase is responsible for the reduction of a ribonucleotide into a deoxyribonucleotide. So CDP to deoxy-CDP to deoxy-CTP. So you get a decrease in deoxy-CTP, which will enable increased incorporation of gemcitabine into DNA. As I mentioned, gemcitabine has also some additional actions. It also behaves like a ribonucleoside, so it can also be incorporated into RNA. And when we found this was the first time that a compound was shown to be incorporated into RNA and to DNA. When you are talking about RNA, then the substrates for RNA incorporations are CTP and UTP. And you see here again, CTP is can not only be formed into deoxy-CTP, but also into CTP. Interestingly, gemcitabine triphosphate also inhibits the formation of CTP, so the enzyme called CTP synthetase, which leads to a decrease in CTP and an increased enables an increased incorporation of gemcitabine into RNA as well. So that's these are mechanisms which we call self-potentiation of a drug. In one of these cell lines which I just mentioned, we measured the incorporation of the accumulation of gemcitabine triphosphate, and what you see here is we put this as a relative amount. This is the accumulation of gemcitabine triphosphate in the ovarian cancer cell lines, and you can see that in the multilayer cells the incorporation is much lower. In the colon cancer cell lines, we've seen a similar picture in the WIDR, HT29 and SW620. The accumulation of DHCTP is much lower than in the monolayers. I mentioned that CTP synthetase is decreased, and that can be seen here in the concentrations of UTP and CTP. When cells are being treated with gemcitabine, we see an increase in UTP levels in both the monolayers and the multilayers, but we see a decrease in CTP, and this in both the monolayers and the multilayers. That means that you can get an increased incorporation of gemcitabine into RNA as well. Regarding the topoisomerase inhibitors, we also measured the accumulation of this topoisomerase inhibitor, the topotecan, and here we see the accumulation of topotecan into the monolayer cells in the SW20, and here we see the accumulation in the multilayer cells. So this is the monolayer cells. These are the monolayers, and here we have the multilayers for HT29 and for the SW20. Moving to the spheroids. Spheroids is also a three-dimensional model system, and they have a little bit, they're being formed in a different way. And I will discuss how we are using spheroids, but also how we can co-culture spheroids with supporting cells in pancreatic cancer. And these supporting cells are called pancreatic stellate cells. And I will use an abbreviation PSC in the next part of my talk. So I'm going to focus first on the model systems and on the efficacy of the gemcitabine and a group of drugs which were called CMET. So pancreatic stellate cells are cells which support pancreatic cancer cells, and they are supposed to be important in the tumor progression. Pancreatic ductal adenocarcinoma is characterized by its dense stromal microenvironment, and stellate cells are considered to be the cause for desmoplasia, and they secrete growth factor called hepatocyte growth factor. And just remember this name because it's coming back. Normally, pancreatic stellate cells are looking like that. When they are quiescent, they are basically inactive, and they just support the the environment. But when they are activated, they change their structure. Here they are some round cells, but here they spread out and they penetrate into to the tumor. When they are activated, you can get you will see a number of changes in the cell. Here we have schematically, this slide is showing what's happening. A quiescent cell will, when it's activated, it's by soluble factors which are secreted by the pancreatic ductal and acinar cells. Also, pancreatic cancer cells themselves can secrete soluble factors which activate the pancreatic stellate cells. As I mentioned, they will change their structure from a little more or less round form into an activated form. When they are activated, they will secrete extracellular proteins like collagen and fibronectin, and they will also secrete growth factors called the hepatocyte growth factor, which I mentioned, but also vascular endothelial growth factor, which is important for the formation of blood vessels, some immune cytokines and chemokines like IL-1 and IL-6, which in general will inhibit the immune system so that the immune system cannot recognize cancer cells and prevent them from proliferating. These growth factors will stimulate the pancreatic cancer cells so that they can proliferate. This will lead to drug resistance. Very important for the tumor, of course, is that they are able to they enable invasion and metastasis, and these are of course the devastating properties of a tumor. When a tumor is going to metastasize, then it's much more difficult to treat. Subsequently, these pancreatic cancer cells will secrete again growth factors, which will activate the stellate cells again. So we have TGF beta-1, we have platelet-derived growth factor, we have fibroblast growth factor, and these can activate the stellate cells again, and then we have a circle which is of activated stellate cells and cancer cells which will support each other. When you want to treat, our idea was when you want to treat cancer cells, pancreatic cancer cells effectively, you have to disrupt this cycle, either for instance the secretion of growth factors or the secretion of these growth factors. So first what we did is characterize whether the cells which we have isolated are indeed secreting growth factors and do what in the normal grow medium, but these cells will secrete growth factors, and what we do is collect this medium, what we call conditioned medium. We add this conditioned medium to stellate cells, which will actually be isolated from tumors, and subsequently we see that these stellate cells are stimulated. They change their form and the characteristic activated cells. So the stellate cells are subsequently cultured as well, and these stellate cells, they will also excrete growth factors, which we add to the pancreatic ductal adenocarcinoma cells, and we have done this in several model systems. We initially started doing this in monolayer to characterize whether these stellate cells are indeed secreting growth factors which might affect the growth of the pancreatic cancer cells and their drug sensitivity. So the circle is almost round. What are the growth factors? One of the growth factors which I'm going to focus on today is called the c-Met growth factors. We have published, there's a couple of reviews on this. Actually, we just published a review on this in critical review in oncology and hematology. It was just published last week. And this is what the c-Met pathway looks like. c-Met is a growth factor receptor, and this is as quite a few growth factors there is located in the membrane. It has, when it's exposed to hepatocyte growth factor, it will dimerize and subsequently lead to an autophosphorylation. This phosphorylation will stimulate several downstream pathways via GRB2. The RAS pathway will be stimulated, PR3 kinase pathway, the STAT3 pathway, the NF-κB pathway will be stimulated, and they will subsequently stimulate proliferation, transformation, migration by acting as stimulants of DNA synthesis. It can also lead to the autophosphorylation of c-Met can also lead to stimulation of the CBL pathway, but this CBL acts in a different way because it will be taken up by the lysosome. It can either be broken down, but be converted to another growth factor which will stimulate other receptors. So you get again some sort of auto-stimulation. What I will do is focus on the inhibition of this c-Met mediated activation and using some inhibitors of c-Met. Crizotinib is the first generation c-Met inhibitor. There's another compound called divantinib and another compound which doesn't have a name yet, PHA665752. Crizotinib was the first generation. This is it's a c-Met inhibitor, but it is also an ALK EML inhibitor and is being used, for instance, for treatment of non-small cell lung cancer. We have demonstrated earlier that crizotinib is synergistic with gemcitabine in pancreatic cancer cells, but also in syngeneic models. And we now proceeded with looking at some more active inhibitors like divantinib and PHA66, etc. First, the selection of model systems. We have established a number of pancreatic ductal adenocarcinoma cell lines, which we call 1, 2, 5. These are what we call primary cells, and these primary cells have been isolated from patients, and we managed to get them growing and proliferating, and we keep them in the low proliferation rate so that they still resemble the tumor as in the properties as we characterize when they came from the patient themselves. What you see here is the phospho-Met expression of these primary pancreatic model systems, and you will see that this model system number 5, PDAC5, has the highest c-Met expression. We have subsequently isolated what we call stage-specific antigen 4 selected cells, and we did this because they look like pancreatic ductal adenocarcinoma stem cells. So on the right part of this picture, you see actually the same PDAC5 here. You see this is the scale is changed here from going for up to 5, 6 to 40, and here we see the PDAC. So this is the similar expression of PDAC5. When we stimulate this PDAC5 with this stellate, pancreatic stellate condition medium, we see an increase in the c-Met expression. However, when we focus on the selected SSEEA4 cells, we already see that these cells have a higher c-Met, phosphorus c-Met expression, which can be increased even more up to sixfold higher with pancreatic stellate cells. So the hypothesis which I put forward earlier is that hepatocyte growth factor possibly stimulates the phosphorus c-Met expression is valid. I just mentioned hepatocyte growth factor. Indeed, we found that hepatocyte growth factor is increased in pancreatic stellate cells, and that's kind of what you see here. This is the non-stimulated stellate cells, and this is stimulated cells that they have a higher hepatocyte growth factor, which is the explanation for the increased phosphorus c-Met, at least one of the explanations. We have subsequently characterized these cells with fluorescence microscopy and bright field microscopy, which is the two lowest slides. And here we have the pancreatic, the PDAC5 cells are here. These are the PDAC5 cells with medium from the stellate cells. You see more cells. What you can see here is the MET expression, so not the phosphorylated, but the MET itself. You see a higher MET expression when we have stellate cells. The phosphorylation of MET is very low here in the PDAC cells. We see here some more higher expression, and when we merge this is DNA staining. When we merge these two pictures, you see that this is very close to the to the nuclear membrane as well. When we look at these stem cell-like cells, and here with medium, you see there's an increased proliferation, but what can be clearly seen, there's a higher MET expression here in both the stem cell-like cells and the stimulated cells, and the phospho-MET is even much higher, and you can see this here. When we merge this, it's even more higher in the you can see this around in the cytoplasm of the cells. You can see this here in the bright field microscopy as well. So this is the morphological characterization of the cells. So how do they respond to the drugs which were usually used for treatment of pancreatic cancer cells? What that's shown in this slide, what you see here is the growth of normal cells, which is put at 100% for the control. This is the cells which are stimulated with conditioned medium, and this is cells treated with gemcitabine, 50 nanomolar, and this is what happens when the cells are incubated with stimulated with conditioned medium. You see that the sensitivity is completely gone. These cells are growing like normal. When we use a somewhat higher concentration, 125 nanomolar, you see an increased effect, but it's again completely abolished by addition of conditioned medium from stellate cells. When we look at the stem-like cells, we see a similar picture. The cells are relatively sensitive to gemcitabine, but it's completely abolished by addition of conditioned medium, both lower and higher concentrations. However, when we look at the c-Met inhibitors, we see that there is no difference. Remember, these are still monolayer cells, which we cultured with this conditioned medium, and we see that they are relatively the drugs are less sensitive to, the cells are less sensitive to tivantinib than for gemcitabine, but still at 200, 500 nanomolar, which is still a relatively low concentration. But most importantly, when we compare these two bars, we see that the conditioned medium will not affect the sensitivity to tivantinib, which is not so strange because the conditioned medium will stimulate here phosphomet, and more phosphomet means that cells are more sensitive to c-MET inhibitor. The stem cell-like actually are less sensitive to these. Oh, here I was going too fast. To tivantinib, although this is not really significant, but most importantly, we see that the conditioned medium will not affect the sensitivity to this inhibitor tivantinib. We did similar experiments for a drug, the new drug called PHA, and here again we see that addition of the conditioned medium will not interfere with the sensitivity to PHA 66. But here we see that the stem-like cells are less sensitive to this drug, and that's generally a property of stem-like cells. They are less sensitive to most drugs, and that's what can be seen here on this slide as well. This compared with this, but again we see that the sensitivity is not affected by the addition of conditioned medium. We can conclude that the c-MET plays a role in drug resistance in pancreatic ductal adenocarcinoma. Moving from the monolayers to the spheroid cells, this slide shows the normal culture conditions in the monolayer. We have a well where the cells are structured as a monolayer, but in the three-dimensional spheroid model, we have cells which are in the spheroid system, and we can also combine two types of cells. We call them here cell A and cell B, and they are mixing, and you can get cells which are in a three-dimensional model system, and at some point they are getting more dense. This is shown in this next slide, in which we showed the overview of the spheroids, and these spheroids can be cultured in also in 96 well plates, which makes it easier to do a large group of experiments. And what you see here is PDAC cells, PDAC5 cells alone, and we see here a lot of different wells, and this is to show the reproducibility of the culture. These are the stem-like cells, and you may not see a lot of difference, but they are a little bit less dense compared to the PDAC cells themselves. Here we have a mixture of PDAC5 cells with stem and or with the stellate cells, and what you see here, this is 10,000 cells, and these are 20,000 cells of the PDAC5 stem-like cells, and here we have a higher amount of stellate cells. And what you can see is when you compare these spheroids with these, that these are more dense than these spheroids, and that can be seen a little bit more clear in this picture, where we amplified some of these spheroid pictures. Here we have the stem-like cells of the PDAC5 themselves. Here we have a co-culture, and you see there are dense, and when we have more stellate cells, they're even more dense. You can easily be seen by these are more dark than these. This is after 96 hours, so that's also a reasonable time to do these experiments. However, you see in these cells, everything is black. And what you would like to know is whether you can see both types of cells. To solve this problem, we labeled the pancreatic stellate with green fluorescent protein, and for the stem-like cells of PDAC5, we modify them by transfecting them with firefly luciferase, and this will have another label called CFP, and these have been adapted to low to serum-free conditions to form these spheroids. How does this firefly work? When we add luciferin and the ATP, which is present in the cells themselves, of course, the firefly luciferase will generate light because it will form oxyluciferin, and that's what we can see under the microscope. That's shown in the next slide. We did this by confocal imaging of the co-cultured spheroids, and what you see here in blue are the stem-like cells after 24 hours, and in green the pancreatic stellate cells. So that the cells, so this is the cyanofluorescent protein, this is blue, and this is the green fluorescent, which is green. This is after 24 hours, and after 48 hours we see this picture. And what we noticed is that the stellate cells are predominantly on the outside of the spheroids, and that the tumor cells are concentrating in the middle part. As I mentioned in 2 slides before, it seems that the spheroids are more dense, and that's what we have been measuring as well. And the more stellate cells we add, and this is the ratio of stellate cells and tumor cells, and what we see here is the density of the tumor cells. Here you can see the ratio 1:8, 1:4, 1:2, 1:1. So we have a higher amount of stellate cells here than here, and we see an increase in the pancreatic tumor cells. How do these cells behave when we expose them to several drugs? Going back to gemcitabine, we co-cultured the stellate cells with the stem cell-like pancreatic cells and exposed them to gemcitabine, and noticed that we had to use a much higher concentration of gemcitabine, exposed it to 5 micromolar gemcitabine. And what you already can see in this slide is that the density of the cells is less than in the previous pictures. When we quantify this, then we see that the cells are not sensitive to gemcitabine. Here we see the control, depending on the amount of stellate cells, 5, 10, or 20,000 cells. And we see here that the higher the number of stellate cells we add, that the cells are growing faster, even when we expose them to 5 micromolar. Even at 10 micromolar, which is really sky high for this drug, and only when we use 50 micromolar, we see that there's a growth inhibition which is more pronounced at the lower number of stellate cells than at the higher number of stellate cells. This means that stellate cells, the more stellate cells are in the neighborhood of a tumor, that we have a higher extent of resistance. We ask ourselves, can we modify the sensitivity, and what is the sensitivity to the other drugs? First, to some other drugs. The reason why I show oxaliplatin is because that's part of the combination of FOLFIRINOX, And what you see here, the IC50 for oxaliplatin is at around 50 nanomolar. And usually for normal pancreatic cells, it's about 5 nanomolar. So this would be somewhere like this. Tivantinib, this is micromolar. You may remember that for the monolayer cells, the sensitivity was also in the low nanomolar range. And crizotinib is also here for the monolayers in the lower micromolar range. The same holds for the PSA, which you may remember from one of the previous slides, was also in around 2 or 3 micromolar. So these three-dimensional cells are insensitive to cytotoxic drugs like oxaliplatin and gemcitabine, as in the previous slide, but also for the c-Met inhibitors. And that the sensitivity is even decreased when you have more stellate cells like these dashed lines here for oxaliplatin is very clear. We ask ourselves, can we increase the sensitivity? And when we combine these two drugs, as we have found in the past for gemcitabine and crizotinib, and again here this is gemcitabine alone. This is the stellate cells alone. They are a little bit sensitive, but this is actually, although significant, this is not relevant. When we add stellate cells, there's no sensitivity at all. However, when we look at tivantinib, the c-Met inhibitor, we see that there's a 50% growth inhibition, which is even better when we have a combination with the stellate cells. And when we combine gemcitabine with tivantinib, we see an increased sensitivity. And especially in the combination with the stellate cells, you see that this bar is lower than this bar, and this difference is significant. This bar is lower, but it's not very relevant. We used a computer model to characterize the interaction between these two drugs, and we found that there's indeed a synergism. As I mentioned, for the pancreatic cells themselves, there is not much synergism. We used Chow-Talalay median drug effect analysis for this calculation. And in this model system, a combination index is being calculated. And when this is around 1, it's not synergistic. And here we see some synergism with a CI value of 0.6, 0.7. However, in the combination culture of stellate cells and pancreatic cancer cells, we see more synergism, and that's around 0.3. This brings me close to the end of my talk. I'm going to give you some summarizing statements and how to proceed in the future. What I wanted to show you is that the tumor cells behave completely different when they are co-cultured with their supporting cells, and in this case we used pancreatic ductal adenocarcinoma cells in combination with their supporting cells, which is called pancreatic stellate cells. They induce resistance even under monolayer conditions. The stellate cells secrete medium containing, for instance, hepatocyte growth factor. That's the growth factor which I showed. And this growth factor is higher in stimulated cells than in non-stimulated cells. And when we culture the pancreatic stellate adenocarcinoma cells in combination with the stellate cells, we see that it's resistant to standard medication consisting of gemcitabine. The co-culture cells could be made visible by using luminescent spheroids and cells labeled with green fluorescent protein. What we also showed is that c-Met might increase the sensitivity to gemcitabine, because you may remember that gemcitabine was ineffective at all, but the c-Met inhibitor increased the sensitivity. As I mentioned, I only focused on the hepatocyte growth factor, and we are now looking at the activation of this MET pathway by using different targets like short hairpin RNA to have a specific inhibition of c-MET, and that's a way to simulate the c-MET phosphate pathway. But we also intend to look at neutralization of this hepatocyte growth factor, because when the hepatocyte growth factor is inactive, then the sensitivity will be increased. We expect that there will be heterogeneity of these pancreatic stellate cells in patients. We have seen some evidence for this when we're looking at the c-MET expression in a different population, and in this way we can determine which subpopulations of patients would benefit from this drug, because we expect that, because when you don't have a phosphor c-MET expression, then the drug will not work. Obviously, I didn't do this work by myself. I contributed, but most of the work was done by one of our PhD students called Pai J, and we had a scientist from Iran who spent a couple of 1 year in Amsterdam called Omid Ferruzi. Another PhD student contributed considerably to this study called Tissam Al-Hassouni. Another postdoc, Ilaria Cannavale, did some work on this together with a postdoc from the University of Leiden. Kay Smith was one of our technicians who was indispensable for a lot of this work. And actually the final responsible person is Elisa Giovanetti, who has taken over some of my, actually most of my supervision work. And we do most of the supervision now together. And that concludes my talk. I thank you all for your attention, and I'm happy to answer any questions.
Thank you, Fritz. Thank you very much for a big, what you call your drug resistance and spheroidal studies. So audience, if you have any questions, please ask, and you have any clarifications, let me know, because Fritz works a lot of areas, and this was one such topic that he works on, so that's how he spoke about these things. So but his area of research also involves a lot of preclinical studies also on cancer drugs.
So I hope it's clear. I understand that the audience was a little bit mixed, pharmacist, chemist,
yes.
biologist.
Yes,
so that's why I took a little bit more time to explain some of the biological systems. And but if there's any unclarity, please let me know.
Yeah. So all these studies, what Fritz spoke about, can be used in all your research work. If you are doing new drug discoveries, if you are doing formulation studies, all these aspects can be used actually. So Moshmi, you can ask.
I put it in the chat box actually. I had a question regarding this 3D studies which he had shown in the plates.
Yeah.
So up to what level those parameters have been studied? Are these validated assays?
You mean which parameters we have studied, or
the 3D models, 3D models study which you presented?
Yes.
Are these continuously used in all your studies, or was it a part of the study which you had presented?
No, they use regularly.
Regularly.
Yes.
This what I showed is that we have a long-term interest in three-dimensional models. The V-bottom models were developed already in the 90s, and the last I would say 10 years we moved to spheroids. And what I mentioned is what I showed are spheroids for pancreatic cancer. But last year we also published a paper on spheroids in non-small cell lung cancer, and 2 years ago another paper on spheroids in glioblastoma. So non-small cell lung cancer is another area of our interest, and the glioblastoma is basically an interest of one of my colleagues, Peter Smilia. And actually the PhD student which I mentioned, PPC, is doing a joint project with my colleague in radiotherapy, and they are looking also at the sensitivity of these spheroids to radiation.
Okay.
So you can use spheroids for quite a lot of different type of tumors. Of course, what I showed you are the spheroids which we managed to establish, and some cell lines are more difficult to form spheroids than other cell lines. You have to be honest on this. But that is also found in vivo, where, for instance, we have for a breast cancer cell line called MCF7 is quite complicated to form three-dimensional model systems. But when we put these cells into a mouse, then we see that the tumor of this cell line is also not very solid. It's quite loose, and that seems to be related to each other.
Okay. We have another question from Dr. Khushwant. You can ask him directly.
Khushwant, do you want to ask? Basically, he's asking about formulation about nanoparticles. I don't think Fritz has worked on these aspects. But we can always try in our lab. Khushwant, as long as you get some money for research.
Actually,
we have tried both.
Just regarding nanoparticles, what PPI is currently actually doing is looking at nanoparticles, the effect of nanoparticles in glioma model systems. That was one of the things which you started with, and she is also going to look at these nanoparticles in pancreatic cancer cells. And I have a collaboration with a student and we published a paper on this review paper some years ago in a student in Leiden, who is combining nanoparticles in the nanoparticles standard drugs with tyrosine kinase targeted drugs, and more importantly, he is also including stimulants of the immune system in these nanoparticles. And that he is actually very popular. He is combining these nanoparticles with vaccination in his model systems. So it, but that was in vivo, but you can use the nanoparticles in these spheroids as well. And of course, it depends on the type of nanoparticles.
Okay, Fritz, I think you have. I think anyone has any. Okay. Okay. Thank you, Fritz. Thank you very much for accepting our invitation, and finally, we could meet more virtually. And we would love to hear some more research, because I always keep mentioning to my colleagues about you. So today they had the opportunity to listen to you and your research, which has been like more than nearing 5 decades, I think. So
not
at
that,
least four and a half decades.
not that old.
Yeah. So thank you very much, Fritz. Thanks a lot for your time.
Yes, thanks for the nice invitation to The last time we spoke, we had snow outside. Now we have the sun shining.
Sun shining, okay.
In 2 weeks we have we had a more than 25 degrees difference in temperature. That's it's not so warm as in India, but it's quite pleasant now.
Thank you so much.
Thank you.
Thank you for helping us, become aware about PDAC and all the 3D drug models which you have been working on. It was really great information, new information for us, most of it.
It was a pleasure to share this.
Yeah. And your willingness to come on this platform and present your research is really helpful for most of us who are sitting, and would have been only virtual platform that we would be able to catch up. Of course, it is morning for you, but it was last lecture of yours for today. And and we are ending today's session with your lecture, and tomorrow we are going to meet at 10:30 again for the second day of the workshop, where we are going to focus on grant application writing, its strength, do's and don'ts with Dr. Patravale from ICT. Till then, bye-bye to everyone and have a great evening.
No, Maushmi, the link for tomorrow is sent separately.
Yeah, it has been shared, sir, everyone.
Okay, thank you.
Thank you so much.
Thank you.
Thank you.
Thank you, Fritz. Okay.
Yeah, that's so tomorrow it starts at 10:30 Indian time.
Yeah.
I will see because 10:30 Indian time is middle of the night.
Okay.
So I may join you a little bit later.
No problem.
Yeah. Thank you again, and see you. You managed to change your manuscript to the proofs, Major?
Yes, Fritz, it's done.
Okay.
All the corrections you had suggested has been done.
That's good, because they were not so. They were relatively minor. I've seen. Okay.
Yeah.
Okay, Fritz. Thank you.
Bye.
Yeah. Goodbye.
Bye.
So we'll break. Okay. Meet tomorrow. Thank you all.